Babe Ruth.
Good morning. Hopefully everyone is caffeinated and had a chance to get some breakfast. We are going to officially kick things off. For those of you who joined us last night for the welcome reception, welcome back. For those of you who did not and just arrived this morning, welcome. We are so pleased to have you all here for our 13th annual Correa Patient and Caregiver meeting here in Tampa, co-hosted with our amazing partners at Moffitt Cancer Center. My name is Kyleigh LiPira. I'm the CEO of the MRF. I have been with the foundation since 2015, and prior to that, I was at Johns Hopkins in Moffitt Cancer Center for a little over 10 years. So really pleased to be with you all today. Really grateful that you are carving out time of your schedules to join with us, to learn, to meet each other, and to grow. I was asked earlier if we were going to do an April Fool's joke. We are not, because I am not good at them. But we're really happy to have you all here today. I'm going to give a brief presentation about the MRF. All right. Is that the April Fool's joke? So I'm going to give a brief presentation on the Melanoma Research Foundation. But before I do that, I just want to point out some resources, a little bit of housekeeping before we get started. I want to identify the Melanoma Research Foundation staff who are here to support you guys this weekend. If you have questions, if you're interested in specific things. So first, I'm going to start off with Eric Krause at the back. Eric is our education associate. He supports all of our educational programming. So if you have questions about webinars or education materials or getting education materials, Eric is a wonderful resource and can help you with that. Kim Watkins, who you're going to meet later, is our advocacy officer. She runs our national advocacy program on the federal and state level. She'll be speaking with you all a little bit later about how you can use your voice in the rare melanoma space to benefit patients and the wider melanoma community. Adam Smart, who is our communications officer. Adam is... Might be a battery issue. Adam is our communications officer. He's going to be taking testimonials that we're going to be sharing during Melanoma Awareness Month in front of that big step and repeat that you see outside this door. We do have a consent form that you'll have to sign, but would love to share your story to the wider melanoma community during the month of May. Janelle Daly. Janelle is our logistics coordinator. Janelle makes all of this wonderful stuff happen. If you have any logistics questions, Janelle is your woman, and she would be happy to answer anything or help you in that regard. Dr. Sara Selig, who you met last night, who you'll meet again, is the director of the Cure program. And Jess Allison, last but not least, is our director of corporate relations. So all of our team here is ready to help and support you this weekend if you all need anything. So with that, we just have a couple other very small housekeeping items. We will be doing luminaries to honor someone who has passed or honor a survivor, and that is the table directly outside. You'll see bags and markers where you can decorate a luminary, and then we'll be displaying those all before dinner tonight. So if you have a minute during a break and you want to get your creative juices and get some markers out, we welcome you to do so. So with that, we are going to kick things off. Again, if at any time you feel overwhelmed or want to have a quiet moment, we have a lobby area out there. We also have our breakout rooms where we're going to be having the psychosocial meetings, and you can grab a snack, grab a coffee, go in there if you feel the need. So first, none of this would be possible without our sponsors. We have many different partners, and many of them are here today, so I hope that you will have a chance to meet them, who support not only this meeting, but our wider Patient Symposium series. So with that, I'd like to tell you a little bit about who the Melanoma Research Foundation is. I was asked that this morning by someone as well, and I want to give you a little overview about the MRF. So we were started about 26 years ago, a little over 26 years ago, by a patient, a young woman who was dealing with her third recurrence of metastatic melanoma. And at the time, there were very few treatment options available. And she was told that at 36, that this was the end of her treatment journey, and there was nothing else that could be done for her. And out of her will and sheer determination, she decided that that answer was not good enough, and she started fundraising with her friends and family and came together to fundraise for the first research grant and form the Melanoma Research Foundation. So the first grant was given out in 1998, and over the many years since then, we have 19 FDA-approved drugs, of which the first drug for metastatic uveal melanoma came on market just last year. So a lot of progress in those 26 years, but it started with the determination of one woman who really saw a gap and a need to help more people. So today, our mission has changed a little bit. We started off with just funding research because there was nothing. But today we educate patients and caregivers and the general public about all forms of melanoma. We provide them with resources to help them along their journey, and we advocate for them on a state and federal level. And our footprint is large. We are a national foundation, so we have advocates in many states across the United States who are advocating involved, whether they're involved with one of our Miles for Melanoma 5K runs, whether they're joining us to have a listening session at the FDA, or participating in our annual Hill Day, or again, coming to a patient symposium. We really work with the entire community. So I'm just going to touch a little bit briefly on our research program, and Sara's going to drill down more into the Cure initiative, but very broadly, and this is the first arm of our mission, we fund research. So as I mentioned, $23 million to date, but one of the things I think is really important about what the MRF does is we involve patients and caregivers in our grant review process. So I would welcome any of you, if you are interested, we welcome your participation in reviewing our grants and providing your opinion on if the science is good. And I will tell you that it sounds intimidating, but many of the scientific advisory committee members change their scores based on the feedback from our advocates. We also have scientific initiatives, and those range, and you can learn more about those. We have scientific meetings and workshops. Again, we have an ocular melanoma science meeting, and many of the clinicians that you're going to hear from today also participate in that meeting, as well as a breakthrough consortium where we bring together sites and centers of excellence across the country to work on specific research projects. Our education program, again, this is what you're attending today. This is one of our education programs. But we have many, many different resources. We have materials. These are all free of charge. You can go onto our website. You can request them. We will mail them to you. You can bring them to your doctor's office. You can bring them to a community event. You can bring them to a school. We also have animated melanoma patient videos, and as well as our Ask the Experts series. In terms of our advocacy program, and as I mentioned, we just had our advocacy days in Washington, D.C., and I would strongly suggest if you're interested in advocacy, please talk to Kim. It's a wonderful event where you can use your voice. You talk to Michelle. Michelle was there as well, talking to her lawmakers. But we have 4 main areas that we focus on, which is research, and that's getting funding through the federal government, through the Department of Defense, and they just did a carve-out for rare disease. So this is really important, and Kim's going to talk more about this, but this is the first time that rare disease was included in that $40 million line item. And I heard through the grapevine that we got a lot of great proposals for uveal melanoma, so that's really promising. Access to treatments, access to telehealth, clinical trial diversity, those are all within our access. Education, so we want our advocates to be educated on the issues, and that's part of what we do for our Hill Day. So when you come, you will have a full day of learning and getting training on the issues that matter the most. Prevention, again, this is banning the tan and allowing sunscreen in schools. We want to ban tanning beds for anyone under 18 across the entire United States. So a federal FDA ban. Right now it's a state-by-state issue, and as we say, we know how kids like to forge their parents' signatures, so we don't know how much that's actually being regulated. And lastly, one of the other areas, it's not a part of our pillars, but it's our prevention and awareness campaigns, and we have many. You will probably have seen our #GetNaked campaign. You'll see our new campaign that's rolling out in May during Melanoma Awareness Month. But this is really creating awareness about checking your skin and seeing a dermatologist every year for full body screening. We have a program for pediatric melanoma, although rare, about 400 children a year are diagnosed with melanoma. And what we want to do is empower the pediatricians, which are often the ones that see the melanoma first. For ocular melanoma, we know that we can't prevent it, but we can create awareness, and we can create awareness about eye health, encouraging everyone to get a dilated eye exam every single year. And lastly, our mucosal melanoma program, Out of the Shadows, because this is a disease that is often diagnosed in later stages, and it's found in places that are not exposed to UV light. So with that, I'm going to share our social media channels. We would love for you all to engage with us through the course of the weekend. And let me go back so you guys can see that. Tag us on social media. We have CureOM channels as well on Facebook and Twitter that are specific for CureOM. So please tag us, use our hashtags, get involved. And again, we really want to kind of see this weekend through your eyes, and we welcome all feedback. So with that, I'm going to end on the MRF, but before I do, I want to welcome one other person who is new to the MRF team. It has not been announced yet to our broader community, but it's been announced internally. I want to introduce Miriam Kadish. Miriam, can you stand up? Miriam is joining the MRF team in April. She's currently at Thomas Jefferson University. She's a licensed social worker, and she is our new Patient Engagement and Education Officer. So please join me in welcoming Miriam. We are very excited to have Miriam join us next month, and you will get to spend time with Miriam today in some of the psychosocial breakouts. So with that, I am very pleased to introduce you all. Many of you met Sara last night, many of you know Sara. But Dr. Sara Selig, who's the director of the CureOM Initiative, and she's going to walk you all through a little bit more about what the CureOM Initiative is and some of the programs.
Thank you, Kyleigh. Can you all hear me okay? I was told last night I was not speaking into the microphone enough.
By your father.
By my father, actually. I was going like this in the back of the room, and you didn't see me. Sorry, Dad. So some of this will be a little bit repetitive for those of you who were here last night, so bear with me. But so pleased to see so many of you in this room, and for those of you who have heard me welcome you before, you know that this typically is a very emotional moment for me. Apologies. I think back to when my husband Greg was first diagnosed, and how alone we felt, and to see this community every year. And this is the first meeting I've been to since COVID, so to be back in person and to feel the community in this room is always so touching and emotional for me, and I hope it's meaningful for all of you to feel the connection with patients and caregivers who, all of our journeys are different, but we sort of understand what it means and what it feels like. And so I'll just echo a little bit of what Kyleigh said. I think sometimes these meetings can feel overwhelming in a number of different ways, and I would just encourage all of you to pace yourselves. All of these sessions in this room are going to be recorded, so if you need to step out for a minute, or if you meet someone and you have a great connection and you just want to continue the conversation, we have this space out here. There's a little patio area, and if you need to come back and look at the recordings afterwards to sort of let some of the information sink in, just please, everybody can sort of chart their own path through the meeting this weekend, and we hope that you'll find benefit from the different sessions. But the slide's not.
We're just spinning for a minute.
Okay, so I can talk without the slides. So for those of you who I haven't had the pleasure of meeting, I am Sara Selig. I'm an internal medicine physician based in Boston. I got into the ocular melanoma field. Now you can tell if you didn't know before, when my husband Greg was diagnosed in 2006. I was a fourth-year medical student and had never heard the terms ocular melanoma used together, probably like many of you in this room. And it was a very scary time. Felt very isolating. There was not a website we could go to. There were no patient resources. And some of the clinicians we were working with told us that they also felt isolated and alone. And they wanted to be able to help us more than they could. And the ocular oncologists we met with said they hadn't met the medical oncologist. And the medical oncologist said they weren't talking with the researchers. And so Greg and I went to work advocating for him And as we did that, we decided that we could lend that energy to the community as a whole. And those were sort of the early roots, if you will, of what has become CureOM. And I think one of the things that always feels emotional at these meetings is that it was my parents and Greg's parents and our siblings putting the packets together at the early meetings. And so it's amazing to have this strong MRF team here now. So we don't really need slides, but as Kyleigh mentioned, the MRF has a tripartite mission, and we've really tried to mirror the CureOM initiative with that tripartite mission. And so I think the backbone, I think so.
He's the man.
Yeah. And as you can see now, I'm very grateful for the MRF team for many reasons. I think that happened on purpose, just so we could see the amazing teamwork within the organization these days. Although my dad still keeps me on my toes, as I mentioned. So this is a picture of Greg and me, and I sort of went through our early journey. And then this is a quote. I shared this last night too, but this is actually from Keith Flaherty, who's a medical oncologist at MGH. And this was what he said to us. And several of Greg's doctors in those early years told us, you know, what's needed is to bring the researchers and clinicians together so we can all put our brains together and figure out how to cure this disease. I talked about our mission, which really mirrors the larger mission of the MRF. And our footprint today, so these are all pictures of CureOM teams. And although CureOM is relatively small within the MRF, I think we're pretty mighty, and I think these pictures, and some of you can probably find yourselves in these pictures. As all of you know, this is a community none of us wanted to be a part of, and how grateful are we that we have found each other, we can build community, and we can advance research, education, and advocacy in this disease. So the backbone, I think of the backbone of CureOM as our patient education and support resources. And this meeting, which we've held annually in person until COVID hit, and then we had a couple years of virtual meetings, I think of a real cornerstone of bringing the community together, connecting and bringing the latest in medical updates, treatments, research to the community, and having discussions. And I think some really interesting discussions have come out of these meetings, the Vision Registry being one of them, where we're bringing patients, clinicians, researchers, sponsors together to think about what role can each of us play in advancing the field. And then, as Kyleigh mentioned, for the larger MRF, there are a number of different educational resources that you can find online, and all of them have uveal specific resources as well, so webinars, print materials, and we have some of those print materials here today. And then the patient meeting. And I think Kyleigh went over the early detection campaign, so I won't belabor this, but these are all of these initiatives that we're talking to you about today were created by patients and caregivers. And so if you all have ideas about how we can do what we're doing better, or how, maybe a new idea, please bring them to us. We have lots of ways for patients to get involved. Kyleigh mentioned the research grant review committee, which is really exciting, but we have lots of other ways for patients and caregivers to get involved. So if there's something that is kind of nagging at you, some ideas that you have, please come up to one of the staff members this weekend. We'd love to engage you more. And then I'll just mention two things on advocacy specific to CureOM. So as Kyleigh mentioned, FDA listening session, we had the first, right before COVID hit, we had the first rare disease listening session for uveal melanoma with the FDA in January or February of 2020. And we're following up with the FDA now on kind of next steps for that. And then we've also had a number of initiatives. One other example of kind of where advocacy and research meet is working with the National Cancer Institute to get uveal melanoma into the Cancer Genome Atlas Project, which has given, I think, the field a really important foundation to build on in the preclinical research space. So this is a quote, maybe I'll just pause and read, if I can. This is from Greg's talk at one of the very early meetings. I think this might have been at the, yeah, it says at the first International Ocular Melanoma Scientific Meeting. And I think this really is still our true north in terms of what we're trying to accomplish in the research space. So Greg said, we have a vision. We want a team of experts to focus on ocular melanoma. The more minds we can bring together to look at OM, the better result we might have. The team, no matter what academic halls they walk, approaches they use, research they are pursuing, are willing to collaborate, push the limits, take risks, think outside the box, be aggressive yet safe, be passionate and integrative. And so I think we, are taking Greg's words with us as we move forward to advance the field. And towards that mission, as you heard last night from Kyleigh this morning, the Cure OM initiative has awarded over $2 million in research grants specific to uveal melanoma. We've launched the Vision Registry, which some of you heard about last night. And if you weren't here for that, please come find me sometime this weekend. We'll read about it in the brochures on your table. Would love to engage many of you more in the Vision Registry efforts. We hold annual global scientific meetings to really increase collaboration and coordination in the research community at a global level. And we have partnerships with the National Cancer Institute, Society for Melanoma Research, Congress, and others. We're always looking for sort of what is the disruptive innovation that we can insert into the research field to really advance research in ocular melanoma. And here are some pictures from some of our scientific meetings over the years. And we've had a few papers published in peer-reviewed journal articles that have come from those meetings. I spoke about the Vision Registry last night a little bit, so I won't belabor this, but I hope all of you in this room have heard about Vision. This is a patient-reported registry where you all are able to enter your own data, and we are centering the patient voice in research, and hopefully we will be able to take ongoing results from this registry to really guide research moving forward. We're hoping that over time the registry can be more and more integrated into other research efforts, into clinical trials, into preclinical research efforts as well. We just published in the fall our first data report from the first year of the registry. I don't think we have the physical copies here, but the report, you can access it online. And if you're having trouble with that, please find one of the MRF staff this weekend and we can help you find it. And if you need help registering for the registry or anything to do with the registry, please ask one of us this weekend. And if we don't have time to dive into it this weekend, I can connect you with Jackie Kraska, who's our research manager, and is happy to work one-on-one with anyone to ease your engagement with Vision. Oh, and you can click, if you go to the melanoma.org, Kyleigh is saying that you can get right to the Vision data report there and just click on it. Thank you for the reminder. So thank you for bearing with me with that whirlwind tour of Cure OM. If anything in there piqued your interest that you want to know more about, please reach out to a staff member this weekend. I'm going to end with a video. So we founded Cure OM in the fall of 2011. Greg was able to see us award our very first research grant. And we've made a lot of progress since then. Clearly not enough, and it's not fast enough. And we feel that every day, and so we're pushing forward. But we celebrated our 10-year anniversary a couple years ago, and I'm going to share a video with you that we sort of made at that point, and then we'll launch into a program from there. when Greg was diagnosed with metastatic disease and we were told he had 6 months to live, we were faced with the hardest time in our lives, and we knew we wanted to do everything we could to save his life. I knew I wanted to do everything I could that was in my power to save my life. And when it became clear that I'm going to be able to save his life, but we extend his life and might have the opportunity to save the lives of other people, that became a real driving force for us. So, with a very hard to find each other, and it can be hard for researchers and clinicians to really get a full grasp of what's happening in this disease and really understand the patient experience and patient preferences.
The local support groups are breast cancer support groups or lung cancer support groups, and nobody with the same kind of cancer. And it's not the same. It's not the same because I didn't have treatment options. I didn't have a group of other people to say, well, this is what it's like to navigate life with one eye. I felt very alone. I felt very, that there wasn't anyone around for me to really bounce ideas of what the next step should be, or if I make a misstep, what does that look like? The big moments for me are meeting the other patients and caregivers. Those are huge moments because you walk into the room and you don't have to explain yourself. I don't have to tell them what my journey looks like. I don't have to tell them what it felt like.
The greatest things about CureOM is the ability as a researcher and clinician to have direct networking and communication with patients.
They give important grounding for research, give researchers an opportunity to speak directly to patients and caregivers and hear directly back. And that's an important conversation to have, so that there's a bi-directional flow of information from the community to the bench and back again.
One of my most favorite things is presenting new data at these patient meetings, and to present data to your peers. But when you get the positives of a clinical trial, new and interesting ways that we could treat the cancer to patients is really, really powerful.
Once we started noticing that there were a lot of patients diagnosed in a certain geographic area, at the Atlantic conference, Dr. Olaf actually said, we really need a registry.
It was clear to us when we were trying to do some epidemiology work that we were kind of missing important parts of understanding the disease. Where were they living? What were their lifestyles? We were all very excited that we've recently launched the Vision Registry, which is really a tool to help kind of gather a lot of this information so that we can look and hopefully better be able to help patients navigate through this, the treatment journey. Not only in registry help researchers, we're also hoping registry acts as a tool to even bring patients together.
The Vision Registry is critically important because it gives an opportunity for patients to be involved in the field. From a research important, it will provide critical information about groups and cohorts of patients and might lead to insight in terms of what are the initiating factors, especially environmental factors.
So when I first started treating patients with uveal melanoma, specifically now, when I have conversations with patients who are newly diagnosed, certainly newly diagnosed with advanced disease, the conversation is really complex. There are at any given time 5, 6 treatment options. Again, the conversation is getting more complex, but it just goes to show how much more we know about you now and the different treatments that we're trying to explore. Now more than ever, the clinician, the oncologist who's seeing patients in the clinic, the researcher who's in the lab, and the patients are working together really and push this new, and I think we're going to see really amazing things over the next 10 years.
Sara and Greg brought together the researchers and the clinicians for the very first time, and that is big because they're everywhere, they're scattered all over the country, and not one person is going to cure this disease. And I feel like we have gained so much momentum to the point where we're going to find an adjuvant therapy. No longer are patients going to be diagnosed and that just stops awareness. We're going to get there.
So, being at Caroline's today, oh, bittersweet for me. It's great. So to hug people who have been here and are doing wonderfully, and their health is good, and I know they'll be here for years to come. And my hope is that there are more patients like them at every meeting, and that patients are living longer and longer, so that there aren't empty chairs at Thanksgiving dinners, and that's why we do this.
When you first get diagnosed, it just feels overwhelming. It's this big mountain in front of you, and you don't know how to get to the other side. But then when you look and you see somebody who is on the other side, somebody who made it through year 1, made it through year 2, year 3, year 4, oh gosh, now we've got somebody who made it through year 10. That is hope, and that's what we need. We need to spread hope. We need for people to understand that you can live, you can thrive, you can live long. So.
I'm sorry about the internet glitchiness, but that video is also on our website as well, and that was really highlighting 10 years, and I think the one thing that we come out with is hope, and I think a lot of you will find a lot of hope this weekend from the information that you're going to learn from the amazing clinician scientists that are here with you today, and the things that are happening in the lab that will directly translate to benefit patients and their loved ones. So we are very hopeful at the MRF, and we are thrilled to be able to bring the wonderful minds that are working on this problem day in and day out to you this weekend. So with that, I want to introduce Dr. Vernon Sondak. Many of you met him last night. He is our wonderful partner at the Moffitt Cancer Center here in Tampa, and he's really helped us put together a dynamic program for this weekend filled with researchers and clinicians and a multidisciplinary team that's really going to provide some great information to everyone here. Dr. Sondak is the chair of the Department of Cutaneous Oncology at the Moffitt Cancer Center and Research Institute here in Tampa, and we're going to get a tour on Sunday so you can see behind the scenes, which is really exciting. He holds the Richard M. Schultz Family Foundation Distinguished Endowed Chair in Cutaneous Oncology and is also a professor in the Departments of Oncologic Sciences and Surgery at the University of South Florida. Since 2004, when he came to Tampa, the Cutaneous Oncology Clinic at Moffitt has grown into one of the largest multidisciplinary treatment centers for pediatric melanoma, which I talked about earlier, and related conditions in the world. And we have Dr. Sondak's full bio in the program, but please join me in officially welcoming him.
Thank you, and thank you all for coming to Tampa and joining us here. First of all, Tampa Bay is really growing dramatically, has grown enormously in the 19 years that I've been here. Just last week was my 90th anniversary when I came down here. We've seen tremendous changes. It's one of the fastest growing areas in the country, and I hope if you get a chance, you'll see some of what Tampa Bay has to offer. If you want any suggestions of where to go or what to do, please let me know. I always love to be a tour guide. But as you've heard, not only has the area grown, but our melanoma program at Moffitt Cancer Center has grown, not just for pediatric melanoma, but for all forms of melanoma, into one of the very largest in the world, one of the top 2 or 3 in the world in terms of the number of melanoma patients that we see every year, adult and pediatric, skin, and all other forms of melanoma. And we have seen unbelievable progress in the treatment of skin melanoma, progress I certainly never thought I would see in my career. Transformational studies that have had tremendous impact on the field of how we take care of melanoma patients have been published in all the most prestigious journals and have influence the care of other kinds of cancer besides skin cancers. A dramatic effect across all of oncology. So what we've begun to do over the last few years is really focus on the problem areas that remain. Pediatric melanoma is one. The brain metastases for melanoma is another. And in some ways, one of the toughest hurdles that remain is uveal melanoma, the melanomas that start in the eye and spread from there. And it's been an area that hasn't shown the progress yet that we've seen in skin melanomas and other skin cancers. But a few years ago, with the help of the Melanoma Research Foundation, we decided to attack this with a team of scientists. You've heard over and over again already at this meeting, and it's just begun how important it was to bring people together. We were able to bring researchers from multiple different cancer centers across Florida and even around the country together in a true team of scientists, and their progress has really astounded me. They've made more progress than I ever thought would be possible. They have leveraged this initial little spark that brought them together into major national grants from the National Cancer Institute, from the Department of Defense, from the state of Florida's scientific funding. So we've just been really pleased to see the dramatic research changes. We are seeing clinical progress. We are chipping away at the wall. We've seen the first drug get approved for uveal melanoma. It's not nearly enough, but I think in the next year we'll see 2 more things get FDA approval. Percutaneous hepatic perfusion that Moffitt has worked on since the very, very beginning. And tumor infiltrating lymphocytes, also a huge emphasis of Moffitt's, and we're one of the only places trying to focus that on uveal melanoma and not just exclusively on skin melanomas. So we're seeing the progress. We've seen how long it takes for research to translate into major changes. It was only 2002, 2 years before I left the University of Michigan and came to Tampa, that we first heard of this gene called BRAF. None of us knew what it was. None of us knew what it meant. It took 8 years before that incredible discovery was affecting melanoma patients. But it also affected my journey, and Kate was one of the reasons I wound up here at Moffitt, because I didn't know what BRAF was. I didn't know what it stood for. I didn't know what it meant. I didn't know if it had anything to do with any other genes I'd ever heard of. And so we asked around and found out that at the University of Michigan there was a famous scientist. They said, I don't know if he knows anything about BRAF, but he's the world's leading expert on CRAF, and he works at the Neurosciences Institute. Maybe we could get him to come over and give a lecture to the melanoma team. And we asked him, he said, well, BRAF, CRAF. And sure enough, he taught us all about this. And I remember asking him afterward, I said, we're going to try to put a grant together. Would you help us work on our melanoma patients? And he looked at me and he said, patients? You mean like people, humans? I said, yeah, most of my patients are humans. Absolutely not. They're impossible to work with. I work on yeast chromosomes, and that's it. He walked out of the room. I never saw him again. I don't know whatever happened to him. And 2 years later, I said, I can go somewhere where everybody is working together, not on yeast, not on fruit flies, but on humans. And so it was really exciting to be able to come here to work with scientists whose first thought is not about the test tube or the mouse, but about the patient, that we can eventually take the progress back into the clinic. And we've got our first clinical trial in uveal melanoma at Moffitt that has come specifically from research that started at Moffitt. That's the ultimate testament to the progress of some of the research that we've been able to achieve. And of course, it's just barely the beginning, not nearly enough. So we're excited about the future, but it can't come fast enough. And thank you all for being here again and the opportunity to bring this whole team together.
Thank you, Dr. Sondak. And just putting in an extra plug for Sunday, if you'll see in the program, which we called it Science Sunday, we're going to get to tour some of the Moffitt labs and see some of that research behind the scenes, which I think will be really exciting. It is now my pleasure to introduce Mr. Nigel Deacon, CureOM Patient and Caregiver Steering Committee member. Nigel is also the co-founder of Ocular Canada, an initiative of Save Your Skin Foundation in Canada. He retired from a career in public education in 2012 when he was diagnosed with metastatic ocular melanoma. And I hope a lot of you are doing the math there. 2012 with metastatic ocular melanoma. So Nigel is sort of a walking beacon of hope, I think, in our community. Nigel now devotes his time to improving outcomes for Canadian ocular melanoma patients. As I mentioned, Nigel is a member of the CureOM and Vision Registry steering committees. So please join me in welcoming Nigel, who's going to speak with us this morning, hopefully about hope in the field. Thank you, Nigel.
Thank you. I'm wondering how we're doing with technology. If you're very far away over there, can you hear me? Okay. First of all, I would like to welcome everybody. And because I'm from Canada, we'll do that in another language. Bienvenue à tout le monde. We're very aware of the diversity in the room. It's a relatively small crowd for a CureOM symposium. When we last met in person in Raleigh in 2019, I think we had about 150 people. So I think the empty chairs remind us that we've just been through a global pandemic and we have all survived it. And thank you to the doctors, we have many of them here, who became heroes by continuing their work despite all of the challenges and the terror that they must have felt in the early days too. Thank you to them. There will be patients who are newly diagnosed here. If you've been diagnosed with ocular melanoma in the last 6 months, would you raise your hand? Let's see if we have. All right, we have one hand. Welcome to our group. And it's, you know, it's a horrible thing to hear because you won't have even heard of the disease before, most likely. And you won't know anybody who has it. And it is very difficult to find people who have it. So special welcome to you today. There are lots of people in the room who are at various stages of living with the disease. Let me point out that we have Linda O'Brien here. Linda, could you stand for a moment and just wave? And Linda has lived with the disease, I think, for 15 years. Does anybody have the experience of living with it for longer than that? All right, Linda, you are our champion today. We also have Faith Houseman. Please stand, Faith, for a moment. Who has lived with the disease, I think, for 8 years. And so thank you for coming again and again to the symposia. Sometimes I wonder why you do it. But it's amazing that you do, and new patients will be glad to talk to you and find comfort in the fact that you continue to live well with the disease. We have, of course, doctors, we have industry partners here, and caregivers. One of my very good friends, Bernadette Borelli, is here. Bernadette, can you stand for a moment, please? Bernadette was caregiver to her husband, who died, I think, in 2015. So again, what brings you back every time we have a symposium? We're immensely grateful for it and your leadership. It takes a lot of courage to keep coming back. You could get tired, you could be doing other things, but here you are. Thank you for that. So I was diagnosed with ocular melanoma in 2010, June 16. We remember those days in our lives. I had absolutely no symptoms. It was discovered by accident with almost magical properties. I didn't understand what was happening to me. My treatment, brachytherapy, happened really very quickly, and that's still the case in Canada, thank goodness. Usually within a couple of weeks you can have whatever treatment is recommended by your ocular oncologist. It didn't really occur to me at that stage that I needed to be in contact with others. I did think, I wonder if anybody else around here has this disease. But it took me a long, long time to come in contact with another human being who had the disease. I think I was referred to the cancer agency because they were involved in developing the plaque, the radioactive beads for the plaque. And when I spoke to the doctor there, I said, I had already had some free consultations with Dr. Google, and so I asked if there would be any scanning to track the disease, and so he said, we don't actually do scans because there's nothing we can do for you if you develop metastasis. So I accepted that as a relatively good answer at the time, but in 2012, just short of 2 years later, I had some inexplicable and very serious pain that took me to an emergency department and a CT scan that revealed that the cancer had indeed spread. And then I knew that I was really in trouble. And again, I was told by oncologists that there wasn't any help available. And I said, I was born in Ireland, so I'm very stubborn. And I said, but there must be something you can do. And I had several recommendations from Dr. Google and said, could we try this? No, we did that, it doesn't work. Well, what about that? Oh, no, we tried that too, and it doesn't work. So I said, oh, I think I have to go to the States. And so during that year, 2012, I was in Pittsburgh, I was in Philadelphia, and I was in Los Angeles. No holidays in any of those places, but looking for doctors who were genuinely trying to help patients in all kinds of experimental ways. And I did end up with a good idea of what I would try to do. And obviously, that worked fairly well, because here I am. I don't remember exactly when I came across CureOM and its work. I did attend a symposium in the fall of 2013, but I think it was organized by another group. There were several groups active at that time in the States. But over the years I became aware of the powerful work that it did. I became aware of Sara's heroic work in bringing people together and doing all of the things that both Kyleigh and Sara have referenced earlier. And I first attended a conference in Washington, DC. And in this slide, you can see me pretending to be American. Because attached to the symposium, the Monday was Hill Day. And I thought that would be amazing to go to the both houses. And so I became a Colorado resident for the day. And we started off strong. I think there were about 7 of us. But there were some younger people in the group who tired rather quickly. And by the end of the day, there were 2 Coloradans left standing. I was one of them. And the other was Carla Tressel, who lived in Colorado, and she died, I think maybe 2 years ago, having contributed enormously to the work of CureOM on the Vision Registry Steering Committee. But it was an amazing day. I really enjoyed it. It's such a powerful idea, how to speak truth to power. I haven't been able to translate it into the Canadian context yet, but I'm working on it. If you have ideas, perhaps you'll help me. I've served on the Patient Caregiver Steering Committee since 2018 and the Vision Platform Steering Committee since 2022. Both Kyleigh and Sara have talked at length about what CureOM does. It has its finger in many pies, and I would just highlight maybe 1 or 2 items. The fundraising by patients is enormously important. Bernadette and Linda, to whom I've already referred, organized a walk here in Florida, and I think we have many people from Florida. Half the group is a local group today. And so if you would be interested in supporting them, their walk will come up again this fall, and they have raised many thousands of dollars for the for CureOM. And even if you're from another part of the country and you don't have a walk, you might want to talk to them as to how you build and how you sustain an important enterprise like that. So I told you at the beginning of my story, it's been going on for quite a long time. And so now I'm going to tell you a little bit about the middle of the story. There really wasn't much around in 2012 when I needed treatment for metastasis, but the clinical trial for the first really effective immunotherapy had closed, and happily as still the case, the company that developed it had a compassionate access program, so I was able to get access to that in the fall of 2012, and I did have some radiation, which is kind of a general go-to treatment that was used more widely back then when there weren't other options. I repeated a course of immunotherapy in 2013, 2014, and 2015. At first it didn't seem to be working at all, and so in January 2013 I was asked to register for hospice, which I did, and that's a very sobering moment. I remember it very, very clearly. But things started to turn around. My immune system found its feet and tackled the cancer, faced it down. And so things got a lot better for me over the next, say, 4 years. I was offered dacarbazine in 2012, and I knew to say no. Makes you very ill, and there's very little benefit. Placebo would be better. Donuts or something like that. But really, you know, since then, there has been an explosion of hope in the ocular melanoma treatment scenario that's beginning to mirror what happened for cutaneous melanoma between, let's say, 2010 and 2015. It's so interesting to see Dr. Orloff, who is a real champion of CureOM, say that her conversations with new metastatic patients are now complex. So that's the most encouraging thing that you can say, that there are lots of different things that can be tried. There is a growing notion that with sequential treatments, carefully chosen, patients can live longer. And at the end of my slide there, you will see reference to adjuvant and neoadjuvant therapy. We never used those words 10 years ago. It didn't seem like it could ever happen. And I was talking with an industry representative last evening who said they were very excited about moving a treatment that they have into the adjuvant setting. So it is happening. I've heard Dr. Shields talk about neoadjuvant therapy and what might help. And there's innovation happening at all levels of this disease, both treatment of primary and treatment of metastatic disease. I can't tell you how encouraging that is. So the first FDA-approved treatment, January of last year, 2022, and approved in Canada, June of 2022. We're always behind. Canadians say it's because we're more careful. I don't believe that. I think they're just not working fast enough. It is an incredible treatment. Have I gone silent? No, I think I can hear. But unhappily, or sadly for us, it's designed in such a way that only half of patients who develop metastasis will benefit from the treatment. It's delivered through a very specific blood marker, HLA-A0201. So you have to have that marker if it's going to work for you. And so wonderful as the treatment is, half of the population can't benefit from it. So we're hoping that that gap will close very quickly because there is a proof of concept. The rest of the task should be easy. I'm no scientist or a doctor. It just should be The pandemic has taken a toll on all of our work, especially patient advocacy work. And so we're starting in many ways from scratch. Some people are discouraged. And so we need to get back in the trenches and be communicating with the patient community. We say that there are 2,000 new patients diagnosed in the States every year. I have no idea why they say that, because nobody knows how many people are diagnosed. We need to be counting them. That's one reason for signing up for the Vision Registry. Sara showed us some numbers last night. So I think there's 430 plus patients signed up. Many of them have not gone on to do the work, which is to, complete the surveys. And I'm not really sure why that's the case, because, my story is 12, 13 years old now, and I did all of the surveys in one sitting in just over an hour. No coffee breaks. So it's very doable, one survey at a time. Within 2 weeks, you have it all done. It can become a place to hold the information where you can go back and you can find it. It's not that daunting. So please sign up. If you've signed up and not completed the surveys, please complete the surveys. It's really important for the patient community that you do that. We don't have Jackie, I think, here today. But you've heard Jackie Kraska's name. She will help anybody who's having difficulty negotiating that journey. And there may be many patients. Do you have the courage to put up your hand if you have registered? All right, so if you can see those hands, you can go to those people and chat a little bit about them. Very important. Outreach to parents, to patients, that's the hard work. So I showed you a slide about the beginning of my OM journey, the middle of my OM journey. Obviously, and I'm telling you about the end of my OM journey, because that continues. For some reason that I don't understand, or anybody else, the cancer went to sleep. So I tried to lead a quiet life in order not to wake it up. My next scans are in next month. No, this month, as of today. And I'm just back from a post-pandemic 3-month trip through South Pacific Islands. And I really enjoyed that. Of course, I kept thinking, I wonder if there's anybody with ocular melanoma around here. That's not the kind of thing you have conversations about at cafes and such. One of the things that happened, though, as I got to know Sara better and listened to her a lot and asked probably too many questions, I wondered how we could build something in Canada to mirror the work that's done by Correa down here. And so first, obviously, I needed a partner. And Canada's leading cutaneous melanoma organization is probably the Xavier Skin Foundation. And it's my pleasure to introduce Jasmine. Jasmine, would you stand up for a moment, please? Jasmine is the new managing director for Xavier Skin Foundation, just settling into the work. And this is one of her first forays into the world of patient advocacy work, and in particular for ocular melanoma. You can see our website there. We're very focused on day-to-day contact with patients, especially newly diagnosed patients. We try to find them when they've just returned from their ocular oncologist with the bad news. And it's amazing that, more and more that is catching on. We'll spend hours chatting with them over the phone, video calls, etc. That piece of the work, I think, has been really difficult for CureOM for the last little while, but I feel confident that it will take off. This is the first step in rebuilding contact, direct contact with patients who need help the most. And so that concludes my remarks. I am filled with hope. I think that there are many, many new treatments in the pipeline. We'll be hearing about some of those today. It's a very long day that we have planned. I won't take up any more of it. One amazing thing that happened for me during the global pandemic was that I was surprised by the arrival of a grandson. I hadn't been expecting that. I'm not sure that his parents were expecting that. But here he is. He's now gone too. Here we are celebrating Christmas together, and I'm the one with the red wine. So thank you for your attention, and on with the program.
Thank you, Nigel, and thank you so much for all of your advocacy and partnership over the years. And I hope many people, if you haven't already had a chance to speak personally with Nigel, will seek him out over the course of the weekend. I think your discussion of the field over the past many years from the patient perspective is a really good segue into our next speaker, Dr. Bill Harbour, who I'm sure in this room needs no introduction. But Dr. Harbour has been a key person in the field of ocular melanoma for many, many years, and has ushered in and guided much of the research that Nigel was referencing that has benefited patients so much. And we're thrilled to have Dr. Harbour with us here today to talk about ocular melanoma past, present, and future. I'll give a quick, brief introduction. You'll have his, no, you can come on up. His full bio, a prestigious bio, is in your program. But for a man who needs no introduction in this room, Dr. Harbour is the chair of the Department of Ophthalmology at University of Texas Southwestern Medical Center, where he continues his clinical practice, educational activities, and research program. His research has been continuously funded for over 20 years by a number of organizations, including the Melanoma Research Foundation, the National Institutes of Health, Research to Prevent Blindness, Alcon Research Institute, and numerous others. And we're so thrilled that you could be here with us today. Thank you, Dr. Harbour.
Thank you, Sara, and thank you all for having me today. And I want to recognize my colleagues here at Moffitt, Dr. Sondak, Dr. Kieran Smalley, Dr. Careth, others, who over the long time that I was at Bascom Palmer, we really developed a lot of relationship both on the clinical care side, sharing the care of many patients with uveal melanoma, and on the research side. So we took advantage of a couple of Florida State grants to really start working together. And this led, one grant led to another, led to a multi-PI R1, which is kind of a big deal. It's 3 different centers, ours, 2, and then the University of Florida. And that's still an ongoing project, which has been very, very fruitful. And I also want to point out Dr. Jose Lusky, who you'll be hearing from later, and Dr. Zelia, who have been partners over the years at Bascom Palmer. And I'm happy to say that even though I've left the great state of Florida, we're still all working together and doing some multi-center, multi-site work together. And just thank you for the opportunity to spend a few minutes with you. And I'm usually asked to talk about the interface between patient care and research, and which I will do. But I was asked to give a talk about sort of an overview of the past, present, and future, which I think about a lot, and I talk about a lot, but I've never really given a talk on it, so it took a while to put this together. And excuse me if some of the slides are not perfect because this is fresh. I didn't like recycle this talk, but it was really fun kind of putting it together. And here are my disclosures. And you know, again, we're talking specifically here about uveal melanoma. We call it ocular melanoma, but there are other kinds of ocular melanoma. There's conjunctival melanoma. There's lid melanomas, things like that. And what's interesting is that we now know that the melanocytes that come into the eye come from a different part of the embryo during development than the ones that go to your skin or to your conjunctiva, and they're wired differently. And that's why you get different kinds of mutations and different kinds of issues arise in the melanoma of the eye. And everybody here knows it's the most common primary eye cancer in adults and the incidence and so forth. So I went back in PubMed. Nobody goes to a real library anymore and pulls out papers from the 1700s, but I went to PubMed, and this was the earliest paper I could find where you could actually find the PDF and look at the article. And the theme here I want you guys to think about is just how far we've come. We think how little has happened, but if you kind of look in a larger context, like how far we've come. First of all, they didn't even realize in the 1800s these were melanomas. They called them sarcomas. And you might ask, well, how did they, how did they even find out they had melanoma? Well, they found out when they went blind. So people didn't go to the ophthalmologist and get their pupils dilated and things like that. If you went blind, you had your eye taken out, and then you would find out sometimes that it's actually a melanoma in your eye. So, that's really the state of affairs until the early 1900s. Equipment got better, and there were direct ophthalmoscopes with a little, how your doctor puts a hat on his head or her head and looks. Well, they used to have a little thing you hold like this, and before there was electricity, they had candles, and they would hold this candle right up to your face and try to look in your eye. So that was kind of the next. I was trying to find a picture of that, and I couldn't. And last night at midnight, I couldn't quite find that picture. But now this is an interesting article. We're jumping forward to 1935 now. They're still calling them sarcomas. But what's interesting, the big question is, do we just enucleate the eye, or do we need to take the entire orbit out? Okay, so that's what an exoneration is when you take the entire orbit out. And the thought was that we just got to get it all out. It was sort of like the concept with radical mastectomy that came up in the early 1900s as well, that we need to radically remove all the cancer and then we can cure the patient, because it's all right there. If we can just get all of it, get around all of it, then we can cure the patient. And so the conclusion was that, if there's any extraocular extension, then we need to do an exoneration. But if there's no extraocular extension, then we can, then an enucleation will be okay. So it was all about anatomy. The cancers are all the same. It's just, did you anatomically get around all of it? And this persisted, but there was a really important paper that came out in 1931 by a British guy named Callender, and a very famous Callender classification resulted from that. And he noticed that if you look under the microscope at uveal melanomas, they look different. Some of them look really spindly, like the one on the left. And others looked what he called epithelioid, these kind of big round cells like the ones on the right. And then there's kind of a mixture in between. And he found that has something to do with those that metastasized and those that didn't. The ones with more epithelioid cells seem to have a higher rate of metastasis than those that didn't. And I think, well, that's kind of that's a different concept, right? If it's all about the anatomy and do we get it all, then why would it matter what cell type it is? So he started planting the seed. It's more than just anatomically getting it all. And this Callender classification is with us to this day. It's been modified over the years. This was a famous reappraisal by a very famous Dr. Zimmerman. And I don't have time to go into all the history, but many of the most famous ophthalmologists over the last 200 years have been involved in studying uveal melanoma. It's occupied the minds of some of the most brilliant people. in ophthalmology. And then, this is interesting, it's kind of an obscure paper. It was actually a PhD thesis, but it was published in an important journal at the time. And this person said, yeah, the epithelioid versus spindle, that seems to be important, but there's also these molecular changes. And back then, they didn't know anything about how to study genes and proteins and things like that. But they had these stains, and there was a stain for reticulin, which is sort of a protein stain. And he found that those that had more reticulin did better than those with less reticulin. We don't really use that today, but it was really the first idea that it's not just the shape of the cells, but it may be that they have different, they're molecularly different too. And I love this little thing at the bottom. I'm going to put this in my next paper and see how well it goes. The results given in the subsequent chapter speak for themselves. And then another very influential concept came along, again, this Dr. Zimmerman. He was a pathologist, a very important pathologist at the time. And he saw there's something called the Armed Forces Institute of Pathology, where back then there were very few ocular pathologists, so many hundreds of these, thousands of eyes were sent to the AFIP, and he and his team would analyze these things. And he noticed that, and of course these are all enucleated eyes. These are, there were no small melanomas in this group. These are all people who probably had vision loss, blindness, etc. So these were more advanced cases. But he noticed that after their enucleation, there was a big increase in metastasis at about 3 to 5 years out, and then it came back down. And he did some sort of pseudo statistics and said, it's the enucleation that's causing the cells to metastasize. It's back to the anatomic model, right? That you squeezed on the eye when you were enucleating, and like toothpaste, you squeezed it into the bloodstream, and that's what caused it to spread. So the prevailing concept was, if you can just get that tumor out, get everything out around it, then you'll prevent it from spreading. But if you have some extraocular extension and you fail to recognize that and you leave it behind, then that's going to spread to the liver. Or if you do an enucleation, you're squeezing it like toothpaste, it squeezes into the bloodstream, and then it goes to the liver. So where does it squeeze out into? There are these big veins that come out of the choroid and into the back. They're called vortex veins. And so for several generations of ophthalmologists and ocular oncologists, whenever you would enucleate, you would first ligate or cut or freeze somehow the vortex veins, because that's where the cells are going to squirt out when you put a little pressure on the eye. So you'd want to clip that off first so they didn't squeeze out into the bloodstream. So it's this very anatomic, sort of simplified concept of how cancer works. So people got really worried about doing enucleations. What do we do here? Does anybody know what movie this is?
Chariots of
Chariots of Fire. Does anybody know how it's related to this story?
The guy who produced it had uveal melanoma.
No, almost, but not quite.
That is true.
I'm sorry, the guy who did what?
The guy who produced this movie had uveal melanoma.
Maybe he did. But one of the guys in the movie was the first to do plaque radiotherapy. So one of the three protagonists, one of them became a missionary, and one of them did something, and then the third protagonist was a guy named Henry Stallard, who went on to become an ophthalmologist, an ocular oncologist, and really developed what we now call plaque radiotherapy. And he used it for both melanoma and retinoblastoma, but it kind of languished for years. He was another British guy over at Moorfields Eye Hospital, and it languished until this Zimmerman hypothesis came up, and people were worried about squeezing the eye when they're doing an enucleation, and so they said, well, maybe we can just put this plaque on here and not squeeze the eye too much, and we'll have better outcomes. So that was really what popularized plaque brachytherapy, and that led eventually to the collaborative ocular melanoma study, which, this was actually controversial at the time, in the 70s and 80s, that, plaque radiotherapy, could it be better than enucleation? Some people said it's better because you're not squeezing on the eye as much. Other people said this is a preposterous, the anatomic model, you got to remove everything. And so they did this comparison. It was actually in three parts, but I'll just focus on the medium-sized tumor part, where they compared plaque radiotherapy with enucleation, and there's absolutely no difference in survival. After 5 years, 10 years, 15 years, there's really no difference in survival. And there's been other studies comparing enucleation to proton beam, comparing it to even resection of the eye. And as long as the treatment is successful, it doesn't really matter what the treatment is, as long as you successfully eradicate the tumor. So it kind of threw into question, well, why are these people, some metastasizing and some are not? If it's not really about the surgery, it's not really about the enucleation necessarily. There was a realization by the end of the 20th century that how you treat the primary tumor didn't seem to really matter as long as it was successful. And the prognostic factors, there were these prognostic factors, the epithelioid cell type, if it was involving the ciliary body, if it had a larger diameter. These were all features that we knew predicted a worse outcome. So it had something to do with the tumor itself, not just about how you did the surgery. At this time, intraocular biopsy was rare. I had the good fortune in my residency to train with a couple of the early pioneers of tumor biopsy, Dr. Jerry Shields and Dr. Jim Augsburger. And they were doing biopsy when nobody else was doing biopsy. And then I did my fellowship at UCSF with Deborah Char, and he was also doing biopsy. And they had accrued 500, 600, 700 patients that had biopsies, but they'd also learned how to do them safely. But at this time, really, very few people were doing biopsy, and many thought it was dangerous and were publicly decrying people who did biopsies. There was no effective treatment for metastasis, as we've heard and we'll hear more about. No standardized surveillance. Most people were told what you were told, that just go home and hope for the best. We're not going to do any testing. Maybe they'll do some liver function tests on your blood work, but there's really nothing we can do, so don't bother. It's a very nihilistic approach, and there was absolutely no industry interest in uveal melanoma. I remember the very first CureOM meeting that Sara had at Dana-Farber, right? Was it? No. Was it Dana? It was Dana-Farber, right, in Boston. And you were gracious to invite me, and you had were able to bring some prominent industry people, from major pharmaceutical companies. And at this time, we had discovered some of the key drivers, the genetic drivers of uveal melanoma, and we're starting to show that there were these different molecular subtypes. Really before they were doing this in more common cancers, we'd already kind of figured this out. And I was trying to talk to these folks, and I won't say what companies they were, but about, maybe we had some ideas about how to target, what we later called class 2 tumors, et cetera. And I remember one of them just told me, look, unless there's a cancer that there's at least 100,000 people a year, no company's going to be interested. I don't care how well defined the cancer is. And I remember telling this person, you're going to find out that lung cancer isn't one cancer. It's going to be many, many cancers, all of which probably have about a few thousand to maybe 10,000 people a year that have them. And of course, that's what's happened. There's many, many subtypes of every cancer type. But there's really zero interest. There was no industry support at any meetings. It was pretty bleak. So there were some hints, again, going back to the differences in the cells, or the tumors, there are differences in tumors rather than just anatomically getting it all. There was this evidence in the early 1990s, being able to look at chromosomes, what we call cytogenetics, But it was noticed that a lot of these tumors were missing a copy of chromosome 3. Often they had extra copies of AQ. This was the first paper, but there were obviously hundreds and hundreds of more papers, and this is still a commonly used method for identifying high risk versus low risk patients. Those with monosomy 3 have a higher risk than those that have 2 copies of chromosome 3. The other concept, and this isn't the only group that did this, but it was a really important paper, and they tried to just use mathematics to say, okay, this is how fast the tumors grow, this is about how fast they're going to get bigger, and here's how big it was when we enucleated it or treated it in whatever way. And if you kind of extrapolate back, that tumor must have, and then when did they metastasize and how long does it take for the metastasis to grow out? That had to have been present before we did the treatment to the eye. The metastasis had to have happened several years before. So this whole concept of micrometastasis came out. And that kind of explains a lot of things, right? It's really not the surgery. As long as it's done properly, the surgery doesn't really alter all that. Whatever, a lot of times when the patient first walks into the ocular oncologist, the die is already set as to whether it's micrometastasized or not. So that changes the mentality from, yes, we need to take good care of the primary tumor and make sure we get it, but that's just the start of the story. It's a systemic problem, not just, oh, did we get it all out of the eye? So, just to characterize this, the concept is that, this micrometastasizes sometime before you enucleate or do plaque radiotherapy or what have you, and then it stays dormant for some period of time, and then for reasons we don't understand, it grows out at some later point. This is also around the time that we started looking at gene expression profiling as a way, the chromosome, the chromosome gains and losses are important and helpful. They don't really tell you much about the biology. So we started looking at the gene expression level and found, that you could divide these tumors into what we now call class 1 and class 2. We found that the bad class 2 tumors actually had mutations in a gene called BAP1, which now there are a number of other cancers that have been shown to be affected by BAP1. There's a lot of work. In fact, the grant that I have with the Moffitt folks is really focused on BAP1 and what loss of BAP1 is doing, how we can target that in cancer. And then, we also found the SF3B1 mutation, which has a less aggressive association with metastasis, but some association. So the more we dig in and get the details, then we can start to ask what drugs might work with this, what approaches, what immunotherapies might target specific subtypes of uveal melanoma. And so we end up with what we know about the landscape now. There are these initiating mutations, GNAQ, GNA11, or some of these more rare ones. But most of the time, you might say, why does uveal melanoma ever happen? You could flip that around and say, why doesn't it always happen? Or why doesn't any cancer always happen? Because just randomly, probably every one of us has a mutation in one of these genes somewhere in our uveal tract. But most of the time, and other mutations as well. It's been shown on the skin that by middle age we have billions of mutations in our skin. But our bodies are pretty good at shutting those down, inducing senescence, killing off the mutant cell. The immune system comes in, sweeps in, and turns it off. So most of the time, it doesn't go anywhere when we get a mutation. We end up with a senescent nevus, or maybe it just goes away completely. But if some other factors happen, and we don't understand all of those factors, it then can progress to uveal melanoma. And the genes that I just mentioned, you end up with class 1 or class 2. Monosomy 3 is typically the class 2 melanomas, and disomy 3 the class 1s. And we can end up stratifying patients into different risks. But we know within that risk category that that's not a uniform group. A few people with class 1 tumors do develop metastasis, for example. So we've been able to go on and stratify this even further using PRAME. PRAME is an oncogene that should only be expressed in the human testis, but it gets re-expressed in a number of different cancers, not just uveal melanoma. And it does a lot of bad things. It seems to mess up the chromosomes, it causes them to grow abnormally, etc. But in uveal melanoma, it allows us to stratify further. So if you're class 1 PRAME negative, you really have a minimal risk of metastasis, and then you go through the different subgroups. And this is kind of what that looks like. This is unpublished, but we're getting very close to publishing this data now. And this is becoming so accurate now that we may be able to use this even to decide how to treat suspicious nevi, people with small tumors that we're not sure if it's truly a full-blown melanoma or not. If it's a class 1 PRAME negative, maybe we can just watch those, for example. So we're preparing another prospective trial to validate that approach. So, we can do all of this now from a single needle biopsy, the class 1, class 2, PRAME, all of the mutations I mentioned, and we're actually working on being able to do the chromosomal gains and losses also off of a single needle biopsy. We can do this because of the platform we developed years ago, which is what's called a microfluidics platform. It takes a very small amount of tissue. We don't have to go in and take a big chunk and cause all kinds of complications. We use artificial intelligence and machine learning 15 years ago when nobody was talking about those things. We were using these techniques, and it's really allowed us to be much more accurate. This has really been all validated by the Collaborative Ocular Oncology Group, which is a group we put together over 15 years ago. It started as about 10 or 12 centers. It's now grown to 25 centers, and this is the group that undertook the Collaborative Ocular Oncology Group 2. Some of your doctors will be on this picture here. And it's turned into, it's now expanded into medical oncology, so that we have our medical oncologist and ocular oncologist in the same room talking about how we can bridge that adjuvant gap, which you're going to be hearing more about today, that adjuvant space between primary treatment of the eye and not waiting for metastatic disease, but engaging the medical oncologist earlier. So this group has been really helpful in moving that along. Just kind of quickly moving along here, you're going to hear a lot more about the next step here. I've talked about how do we use early detection to treat patients earlier, personalized surveillance based on their genetic profile. Patients with high-risk tumors should be considered for adjuvant trials, or at least for elevated surveillance, where somebody with a lower risk tumor doesn't need all of that extra surveillance, so we can personalize people's treatment. You're going to be hearing more about this, so I'm not going to spend a lot of time on it, but in order to be treating patients in the adjuvant space, we need to have some idea that we have a drug that's active against metastatic disease, and we've never had that before. So we talked a lot about adjuvant therapy, and we know these are micrometastasizing. We know which patients probably have micrometastatic disease that we can't see, but what are we going to do about it? So now, Tebentafusp is really the first chink in the armor, right? There's been a lot of bullets thrown at the armor over the years, and none of them have had any effect until now. So this isn't going to be the cure for everything, but it's the first effective therapy. And I think, first of all, it shows that it's possible to treat metastatic uveal melanoma. Number two, because uveal melanoma didn't respond well to checkpoint inhibitors, people kind of threw up their hands and said, oh, this is immunologically cold tumor, it can't be addressed immunologically. there's not enough mutations, whatever the reason. But I think this has shown that that's not true. It is possible. Yeah, they don't have a lot of mutations like cutaneous melanomas, but that doesn't mean that you can't take other approaches to immunotherapy. And I don't have time to go into it today, but the main drivers of metastasis, BAP1 mutations, SF3B1 mutations, and PRAME, they all alter the immune microenvironment within the tumor in different ways. And so the more we understand that, then the more that we can shape immune therapy and other types of therapy based on the subtype of uveal melanoma. This is a proof of principle that it can be effective, as I mentioned, and it's really, I think, opened the floodgate to other pharma companies wanting to get involved in uveal melanoma, both in the immunotherapy space and the targeted therapy space, that it is possible to have an impact on metastatic disease and hopefully in the adjuvant setting as well. PRAME is another target because it's commonly abnormally expressed in other cancer types. That's attracted industry interest, including Immunocore, which is the company that developed Tibinifus. Their second drug is the same type of drug but targeting PRAME. And this is already in clinical trials. So this is another area of potential interest. And then in collaboration with Dr. Lutsky, who will likely talk about this, we found that, maybe before we abandon checkpoint inhibitor therapy altogether in uveal melanoma, we found that, well, one reason checkpoint inhibitor therapy doesn't work is that we're not hitting the right checkpoints. The ones that work in cutaneous melanoma, like CTLA4 and PD1, are hardly expressed in uveal melanoma. But another one, LAG3, which is now very, very hot in the checkpoint inhibitor space, is predominantly expressed in uveal melanoma. So we worked with Bristol Myers Squibb to undertake a trial in metastatic patients, and that trial is still underway, but we'll be getting some results. Just completed, okay. And then the other important thing, I don't want to steal your thunder, but we important in all trials, and I'll mention this with regard to the Immunocore trial, important in all trials going forward is to have correlative biomarkers, that we're looking at, did the drug actually hit target? If it didn't work, why did it not work? So we don't just say, oh, it worked or it didn't work, and if it didn't work, well, we just lost a lot of time and money. We want to know why it didn't work. Did it actually not activate the T cells? Did it actually not get to target? What have you. So we're doing that type of study. And then just the remaining few minutes, it's hard to get on Google and look at anything about cancer or other parts of medicine without running into liquid biopsy. Everybody's obsessed with liquid biopsy this stage, right? So what is the role of liquid biopsy, or what do we know about liquid biopsy in uveal melanoma? Well, first, what liquids might we want to look at? These are the main ones that have been looked at. The vitreous, which is the kind of semi-liquid fluid in the back part of the eye, the aqueous, which is the aqueous humor in the front part of the eye, and blood. So there are others, but I'll focus on these. You say, well, what are you looking for in the liquid? We can look at biomolecules like DNA, RNA, protein. You can look for actual cancer cells, CTC, circulating tumor cells. Or you can look at little particles, little vesicles that are shed from the cancer cells, exosomes. And then they have little cargo inside of them, proteins and things like that. So this is a rapidly expanding space. There's a lot of people looking at different aspects of this. But just real quickly, there's been some interesting work. This by a colleague and also a collaborative ocular oncology group member, Prithvi Murthrunjaya at Stanford and his team. They do their biopsies by first doing a vitrectomy. They go in and take the vitreous out, and then they do the needle biopsy. So they take that opportunity to take the vitreous and look at the proteins in the vitreous. And what they found is by using class 1, class 2, and PRAME as a guideline for looking at protein changes, they have found proteins that are different between, maybe they're present in a PRAME negative and absent in a PRAME positive tumor or so forth. The question there is, if you're already inside the eye, why not just biopsy the tumor? So I think what they're really wanting to do is to move to the aqueous, right? Because if you're biopsying the aqueous, that's a game changer. Because you can do that in the office very easily. It doesn't sound nice or pleasant, but you actually don't feel it at all. One drop to numb the eye, we can take a little fluid out of your aqueous, and you don't even feel it. And we can do it over and over again, right? It's not a one-time thing where you go do a biopsy and it's either A or B. You can do it every 6 months and look for a change over time, things like that. So they're trying to move this, and other groups are trying to move this kind of proteomic approach into the aqueous. You can also look for cell-free DNA in the aqueous, like we do in other eye cancers like retinoblastoma. It doesn't work that well, at least so far. This is a study out of Memorial Sloan Kettering. You actually have to radiate the tumor first to get it to shed DNA. So again, you could ask, well, if I'm already in there doing surgery, why don't I just biopsy the tumor? I don't need to do the aqueous. So we're not really there yet, but I think there's a lot of potential for the aqueous. Now, circulating tumor cells, there have been a number of groups that have looked at this. I think the best work has been Ellen Gray, out of Australia. And they've been able to find circulating melanoma cells in most patients with uveal melanoma. If you remember back what I was saying before about the micrometastasis happening, you can find circulating tumor cells. The question is, if you take 10 cc's of blood and you find 3 tumor cells, and the tumor in the eye has 10 billion cells, are those 3 cells really representative of what's in the eye, or are these actually different, and that's why they're shedding? So I'm not overly optimistic that this is going to be something that we'll be using clinically in the near future. However, I think circulating tumor DNA is really here to stay, not just in uveal melanoma, but in other cancer types. This also was Ellen's early work, where she could show, looking just, for example, at this patient had a GNA11 mutation, GNA11 mutation. You can, if you know exactly what piece of DNA to look for, you're not trying to look for large stretches of DNA, you can actually design a test that's super duper sensitive, that you can find 1 molecule of mutant DNA in 1 million molecules of normal DNA. And that's what she did here. And you can't use it to replace like a tumor biopsy to tell you if you're class 1 or class 2, but you can look for it to start to rise. And that can give you a head start in knowing that metastasis may be developing before it appears on imaging. It can be used the other way as well to see if you're clearing the DNA, the tumor DNA after treatment, whether that be primary treatment or systemic treatment. And that's really what Immunocore found in their study. And I think that's the other big breakthrough from this study. Not only did tebinafusp have a benefit, but it showed the superiority of looking at tumor DNA over imaging and assessing how people are responding to therapy. So the patients who did the best, patients who did the best are the ones who cleared their ctDNA, but these aren't necessarily the ones whose tumors shrank. Some of these actually seem to increase. And it's a phenomenon that we're now referring to as pseudoprogression, where the inflammation may actually cause the lesion to swell on imaging even though the tumor's dying. So this is a really important breakthrough. I think that ctDNA is increasingly going to be a part of the correlative biomarkers that we use in future studies. Finally, this concept of micrometastasis, it's not that this happens one time. This is actually happening all the time, okay? We don't know what the rate is, but cells get in, from Ellen Gray's work and others, we know that cells are getting into the bloodstream all the time. But it's a very inefficient process. Most of these don't survive. The ones that do lay dormant for years, some of them never come out of dormancy. So this isn't like a one-time event and you're trying to catch the patient the day before they micrometastasize. You know, oh, we cured that one, we didn't cure that one because it happened the day after we treated them. It's not really like that. It's like collecting rainwater in a bucket. It takes a long time for that metastasis to successfully take root and grow. So we have to kind of think of metastasis not like a one-time event. It's kind of like a very inefficient process that over time and further mutations in the tumor, it kind of learns how to master its microenvironment. You know, we do know that BAP1 mutations, PRAME, SF3B1 mutations to some extent, what they seem to be doing, they don't make the cells grow faster, they don't make them more invasive, but they somehow are allowing them to successfully colonize the liver or other sites. And that's really where our thinking is now, that these different subtypes, it doesn't cause them to shed more of one type more than another. It allows them to successfully colonize distant sites such as the liver. And probably the most important aspect of that is the immune surveillance. You know, that BAP1 loss is having an effect on immune surveillance. There are certainly other factors as well that are liver-specific factors, that these genetic changes are interacting with factors in the liver, to epigenetic factors, other factors that allow the tumor cells to successfully colonize. So, you know, the new concept of adjuvant or even neoadjuvant therapy, where we would treat systemically even prior to treating the primary tumor, would be to allow us to kill the micrometastatic disease around the same time that we're treating the primary tumor. So this is really the new thinking in the field. Adjuvant therapy or even neoadjuvant therapy, treating around the time of treating the primary tumor. This is just one example of an adjuvant trial that Dr. Lutsky and Dr. Correa and I have, we're about to start, hopefully, in high-risk patients. And there are others that you'll be hearing about from industry that have really good rationale for adjuvant or even neoadjuvant therapy. So, in summary, you know, to really improve survival, we have to get out of this old way of thinking, did we get it all? Did we get around it all? It's really more than that. It's a systemic disease from the beginning. Yes, we have to treat the primary tumor properly to get rid of the source, but then we have to really think about the whole patient, really from the very beginning. We don't wait until there's something shows up in the liver to think it's a systemic disease. And we need to be trying to treat earlier. Primary tumors, if we can detect melanomas from nevi earlier, that would be helpful. Treating in the neoadjuvant to adjuvant setting, pushing the time back. You know, the longer metastasis goes on, the more additional mutations the tumors accumulate and they become more resistant. So the earlier we're able to intervene, either kill the tumor cells or drive them into a senescent dormant state, like yours fell asleep. How do we make all of them fall asleep, right? That's what we want to be able to do. So there's now lots of progress, there's lots of hope, as you're going to hear about today from academia and industry. And we talked about, you know, tumor biopsy and liquid biopsy. So thank you for your time.
Thank you so much, Dr. Harbour. That was an excellent presentation. Sara and I were actually just sitting there saying, wow, we learned so much. It was such a great overview of the field. It was really excellent. So thank you so much for that. We are going to hold questions until the end of our first session this morning. So hold your questions for Dr. Harbour because you will have a chance to ask them. What we're going to do right now is we're going to go into our concurrent sessions. So if you are interested in attending any of the psychosocial sessions, Molly Vicino, she's going to come up and provide instructions to this group. Again, we have 2 concurrent sessions that are going to be happening in the 2 rooms. If you go out this hall, Adam at the back can help guide you out the hall to the right. There will be 2 sessions, one in advanced care planning and one in managing anxiety for patients and caregivers. For those of you staying in this main room, the next 2 talks will be on primary disease, and then again, we'll have Dr. Harbour come back and join the panel for questions. And anyone who's attending the concurrent sessions can also come back and join us here for the Q&A as well. One more housekeeping note. Right before we break for lunch, we will be taking a group photo. So please, if you need to run back up to your rooms, if you could just come back here right before lunch so we can take that photo. We're going to take it at the back of the room where you see that Tampa skyline. So please come back and join us for the photo. And then lastly, one fun fact for you all. Jack Eberts, who is the producer of Jake Eberts, I can't even read my own writing, even though I know his name. Jake Eberts, who is the producer of Chariots of Fire, we actually honored him posthumously at our New York Gala in 2012. He died in September that year, and we are still in touch with his daughter, Wendy. So, fun little fact of how he's connected to Correa. So, I'm now going to introduce Molly, and she's going to come up and give us some instructions. Molly is an oncology social worker at the City Kimmel Cancer Center at TJU in Philadelphia. Prior to working in oncology, she supported hospice and palliative care patients, and both in the community and in hospital settings, and received a certification as an end-of-life doula from the University of Vermont in 2018. She works with the melanoma community at Jefferson and co-facilitates a monthly OM support group that the MRF co-hosts with them. So I'm going to ask Molly to join me, and then again, I just want to let everyone know that to come back in the main room if you want to participate in the Q&A and ask Dr. Harbour or any of the other presenters questions after the concurrent sessions. So, Molly?
Thank you. You really said it all. I don't have much more to add. Well, we're going to break out in one of the first kind of breakout room just out to the right and around the corner. We're going to start talking about advanced care planning, living wills, advanced directives, all of that. And then Miriam is going to take over and talk to us a bit about managing anxiety. Patients and caregivers, welcome. Industry partners, welcome. This is a topic for everyone. MRF staff, welcome. So if you want to take a break from the primary disease content, come and join us. And yeah, we'll be there. You can follow me out.
Great. So we'll just give everyone a couple minutes if you want to head to those sessions or grab a coffee, grab a water. We're just going to get set up for our next session here. So we'll just give everybody a minute.
With everybody, the chef there.
I must have been, I guess. For now, there's nothing to bring Sara out. Really well. Dr. They all did. Dr. Harbour, Dr. Selig. That's it. Yeah. Good for you.
Thank you.
Okay. Thank you.
Well, that's the area. Aaron mentioned that.
If you can grab your seats, we are going to resume with our next speaker. So everyone can have a seat. We are ready to get started. Thank you all. And if you are planning on doing the concurrent sessions, again, those are in the other two smaller rooms. So we are going to get started with our next speaker. All right. Everyone can grab their seats so we can get started so Dr. Williams can present.
You need a whistle.
I do need a whistle. Or a taser or something. All right. So we are going to get started with our second presentation on primary disease, and it's my great pleasure to introduce Dr. Basil Williams. Dr. Williams is a board-certified ophthalmologist and an associate professor of ophthalmology at the Bascom-Palmer Eye Institute at the University of Miami. He is the department's vice chair for diversity, equity, and inclusion and specializes in ocular oncology and vitreoretinal surgery and diseases. I feel like I'm not pronouncing that correctly. Big words. I'm going to ask Dr. Williams to join me at the podium now. Again, we're going to hold all the questions until the end, so we have one more presentation after Dr. Williams, and then we'll bring the entire panel back up here and open the floor to questions. So please join me in welcoming Dr. Williams.
So first, I wanted to say thanks so much for giving me the opportunity to speak with you today. It really is an honor for me to be here. First, from an educational standpoint, you obviously all heard the fantastic talk by Dr. Harbour, and that's not surprising to you. But I did my residency in retina training at Bascom-Palmer. Dr. Harbour was running the Ocular Oncology show. And then after me, Dr. Correa is going to be speaking, and I actually followed her at the University of Cincinnati, where I was in practice for a little while, and then followed her again down to Miami. And so it's an honor to be speaking between the two of them. And also even more of an honor after I met Dr. Selig and we got to talk a little bit at an ocular oncology meeting, I started to hear more about Pure OM and understand what it meant to interact with patients and kind of hear from the different perspective. A lot of times we pay so much attention to the scientific part of things, and I think it's really, really powerful to hear your stories and understand things from your perspective. And I think just kind of the journey over the last few years of being able to participate in Pure OM has changed the way I interact with patients and how I practice. And so I want to thank you all for that as I give this presentation. I also want to say that Dr. Harbour's science was fantastic. My talk is more practical on just kind of the one-on-ones of primary ocular melanoma. I will give a heads up from the beginning. There are some pictures, there are some videos of treatments. So I will give a little warning. If you don't want to look, by all means don't look. But hopefully we can keep your attention. So these are my financial disclosures. And the goals of this talk, what is ocular melanoma? And as Dr. Harbour mentioned, we'll be focusing mostly on uveal melanoma. What are kind of some of the risk factors? How do we diagnose this? And how do we treat it? So we know the most common primary intraocular malignancy. Dr. Harbour kind of mentioned all of this. And there are 3 locations. Iris makes up a small component of the ciliary body, also a small component. But choroid makes up the vast majority in the back part of the eye. Men and women seem to have similar distribution, more or less. This was the demographics, at least in Cincinnati, the youngest being 4 and the oldest being 95. I think either Dr. Correa or Dr. Harbour could correct me on this based on the Miami data, but it's basically a wide range of patients. We know from a racial standpoint, this does seem to affect patients with lighter skin pigmentation, and so more commonly seen in Caucasians. And being now in Florida, you know, we think more about ethnicity on top of race, and we don't have great data on what happens if you are from a Hispanic background, whether it be from an Afro-Hispanic background or more from a white Hispanic background. We don't know that information so well. We do know that there are some risk factors that play a role. And ocular or oculodermal melanocytosis is something that we'll talk about in a second. BAP1 predisposition syndrome we'll talk about a little bit as well. And as you can see in this picture, as a conversation I've had with many patients, you see 2 lesions in the same eye, and many patients ask, well, is it possible for me to get another tumor in this eye or in the other eye? What are the chances of that happening? They're obviously extremely low, but when we think about patients with multiple tumors in each eye, or a very, very young age range, we think about the factors that are more likely to be genetic components, so ocular or oculodermal melanocytosis or a BAP1 predisposition syndrome. We also think about UV light and whether or not that plays a role. Patients all the time are asking, well, should I have worn sunglasses and could that have prevented this? And we really don't see a UV signature in these tumors, and so that seems not to play a large role. Ocular or oculodermal melanocytosis, and so this is where you have patches of the melanocytes involving the skin, the front part of the eye, and the back of the eye as well. And so if you look at this picture here of a patient, you could say, oh, well, the iris is pretty dark in pigmentation, and you might not think much of it. But when you compare to the other eye, you can see that there's a significant difference between the two, and this is what you think of characteristically of the melanocytosis, increased pigmentation there. When you look closer at the iris, you can also see these little mammillations, so these little buds. And you also see this in the palate and sometimes in the meninges as well. We won't talk about how we got that tissue. But when you look at the back part of the eye, you can see that there's diffuse choroidal pigment as well. Again, here's a patient with a melanoma there. But when you look at this, you might say, okay, this is what it's supposed to look like. Again, we'll take a look at the other eye, and this patient does have diabetes, so you can see some of the spots of blood, but you can also tell the difference in color between the 2 eyes. And so the eye that has the darker pigmentation in the background has melanocytosis and an increased risk of melanoma. And so with oculodermal melanocytosis, we know that 1 in 400 Caucasians develop uveal melanoma, so it increases the risk. It also doubles the risk of metastasis. So this is something that we pay attention to when we see this in patients. You can also not have any of the changes in the front part of the eye and just have it in the back part of the eye. And here are the 2 eyes of a patient, and you can see this scan showing the retina and the choroid behind the retina. And when you look at the area in the other eye, you can see that deeper, it's a much thicker area. So that darkened pigmentation also increases the thickness of the choroid. And once again, a separate patient from this paper, and you can see that in this picture on the left eye, the choroid is very, very thin. And you can actually see the line going through the area of pigmentation and then out of the area of pigmentation, and you can see the difference in the thickness of the choroid. So we know that this does increase the risk of tumor development. Some work out of Cincinnati previously with Dr. Correa showed that there seems to be an association with development of uveal melanoma. And these are actually, this is a patient who had a uveal melanoma with melanocytosis and developed a second tumor later on. And similarly here, a patient as well. BAP1 tumor predisposition syndrome. And so this is a syndrome that was discovered somewhat recently, and we know that there's multiple cancers that are associated with this condition. The vast majority of patients develop one or more malignancies by 50 years old, with uveal melanoma being the most common. And so when we have patients who are particularly young, or they have a family history of multiple of these types of cancers, then we have a conversation about evaluation for this condition, because it's important to know for screening for them and for potential family members to know that there is a risk of passing this on to family members as well. And so I think about it for everyone under the age of 40 years old or with a family history of associated cancer, but I think there's kind of a range in how people think about this and kind of working patients up. How do we make this diagnosis? So the collaborative ocular melanoma study showed that ocular oncologists are 99% correct in making the diagnosis. Most of the time we use ancillary testing, so you may wonder, why when I come in do I sit in the office for 1,000,000 hours and have 1,000,000 different tests? And this is why, because this is helpful to make the diagnosis and for following things afterwards. It is a clinical diagnosis. It's one of the few conditions in oncology where you don't have to get tissue to make the diagnosis. But sometimes you do get tissue, and that's particularly helpful for molecular prognosis, which Dr. Correa will talk about, but I'll talk a little bit about biopsy and the role that it plays as well. So some of the characteristics that we see. Melanoma is often a pigmented tumor, but not always. It often has orange pigment over it, or lipofuscin, and subretinal fluid. And so here you can see some of the orange pigmentation, and you can see the fluid that's developed around it. It is a little subtle. When you look at it in autofluorescence, though, it highlights some of that orange pigment and the subretinal fluid. On ultrasound, these lesions tend to be hollow. What does hollow mean? You can see kind of the outline or the borders of it being bright white, and on the inside it tends to be a much darker kind of color, and that helps you identify that as a melanoma. And then again, using an OCT, you can see subretinal fluid, and so that's helpful for some of the subtle lesions. Again, here you can see a lesion, you can see the orange pigmentation, and there's so much fluid actually that there's a fairly large retinal detachment, which you can see on the autofluorescence again, and the ultrasound helps to highlight that. This characteristic, you can see an amelanotic melanoma, so you can't see much in the way of pigment, but the ultrasound shows a classic mushroom shape. And that's because the lesion, as it grows larger, grows from the choroid, breaks through Bruch's membrane, and so you kind of get a squeezing of Bruch's membrane, and you get the apex of that mushroom configuration. You can also see on ultrasound that there's some hollow or darkening, darker area of the lesion on ultrasound at the base. And then sometimes when it breaks through, you can get some bleeding. And then some of the ultrasound features that we talked about already. Choroidal nevus is one of the most common things that we try and differentiate from melanoma. Most of the time it's very easy. This is essentially like a mole that you have on your skin, except it's in the back of your eye. So some studies have shown that it's seen in about 5% of patients of Caucasian background. However, those studies were done when we had old imaging and you couldn't get a wide view of the eye. So it's probably a higher percentage of patients that have this. Usually it's asymptomatic, kind of similar to what Nigel had mentioned. A lot of these lesions are in an area where you wouldn't notice it. And it is rare to have transformation to a melanoma. So Dr. Gass was a very well-known retina specialist at Bascom Palmer, and he came up with some criteria that we use to help differentiate these benign lesions from malignant lesions. And you can see the top three, drusen or features of chronicity and choroidal neovascular membranes, tend to be favorable, meaning that it's more likely to be a benign lesion if you see these characteristics. Whereas as the lesion gets thicker or has orange pigmentation, low reflectivity, meaning kind of like hollow as we were talking about, those tend to be signs more of a melanoma. Since Dr. Gass came up with this, there's been a lot of others who have also talked about this. The Shields group has mentioned 2 fine small ocular melanoma using helpful hints daily and kind of going through the characteristics that help you identify when the lesion is more likely to be a melanoma, and then more recently kind of using imaging characteristics to talk about this as well. In the UK, Bertil D'Amato has talked about the moles classification, again using the same characteristics that we've talked about, And then Arun Singh in Cleveland has a model to try and help us identify the difference between these lesions. And so as you can see, based on the number of modalities or systems that we have to differentiate them, it can be somewhat challenging to differentiate a large-sized nevus and a small melanoma. So I'll go over some of the imaging characteristics of the nevi, of these small lesions. So number one, they tend to have chronic changes, like drusen. And you can see these yellow spots here on the color picture. On the top OCT, you can see those little bumps, which are representative of the drusen. And then on the bottom, you can see the ultrasound shows a thin lesion. Here's a nevus that was followed for a long time, 30+ years. And you can see over time, it develops some areas of chronicity. And so you can see some fibrosis there and some RPE hyperplasia. Basically meaning when we see those characteristics, we know the lesion's been there for a long time and it wasn't likely to be a rapid or new development, therefore less likely to be a melanoma. What about choroidal neovascular membranes? What is this? You see a small lesion here and you see some blood over the top of the lesion. I showed a picture earlier of a melanoma with a lot of blood, and so you may wonder, well, is this a melanoma? In actuality, this is just abnormal blood vessels that are growing from around the area of the tumor underneath the retina and leaking fluid. And this is a sign that the lesion has been there for a long time and is actually a good sign, less likely for this to be a melanoma. And you can see some of the angiography pictures that help demonstrate that as well. And then one other characteristic is a halo. And this is basically due to balloon cell degeneration that can happen around the periphery of a lesion. So this is usually a good prognostic sign as well. Now about the imaging. We take a lot of pictures in ocular oncology, and that helps us differentiate these lesions, but it also helps us do a few other things. I use fundus photography or the pictures of the lesions so I can talk with the patients, show them what I'm seeing on the screen. So that way we can have an idea of what's going on together. So you can see here there's a pigmented lesion superiorly. It was kind of thin, and over time you can see that it grew into a melanoma. This is over a few-year period. Now obviously, if you're just looking at this, you could kind of tell this difference just by seeing it. But it's not always this clear. So here's a little bit of a smaller lesion where you follow it with photos, and you can look in this area, and if you use the blood vessels like fingerprints, you can see that there is growth of this lesion past the blood vessel, and over a small or a short period of time, you can say, oh, okay, nevi may grow some, but they should not grow that much that quickly. And that helps you get an idea that this was likely a melanoma to begin with, and it was just hard to tell in the beginning, but with a short period of observation, you could see the growth. And so the pictures kind of help us identify those small areas. Ultrasound, we've talked about a little bit already. The thickness of the lesions is really helpful. Watching it grow, not only on the pictures, but also on the ultrasound kind of gives you an idea. So again, just by seeing this patient, you can see the area on the top versus the bottom. There's a big difference in terms of the size. The ultrasound also documents that in thickness. And so this was a melanoma, perhaps from the beginning, but we just didn't see those characteristics, and over time you were able to see it. On top, you can see the mushroom-shaped melanoma that we talked about, the characteristic appearance, but ultrasound also helps us manage some patients because here you can see extraocular extension. So you can see that the tumor has already grown beyond the wall of the eye and is behind the eye, and sometimes that helps us differentiate what treatment is required. If it's very, very small, you may still do plaque or proton treatment. If there's a huge area of extraocular extension, it will potentially shift your treatment approach. Here's an ultrasound of 3 different lesions. And when you look at the color pictures, it's helpful to identify some of the differences, but you can see that both for choroidal metastasis and choroidal hemangioma, you don't have that characteristic hollow appearance, so it tends to be pretty bright throughout the entire lesion. Whereas in a fusion, which is usually some component of fluid essentially, you can see that it's extremely dark, much more dark than solid tissue would be. And so you can see how the ultrasound helps us in that scenario. Here's a tumor near the front part of the eye. And the goal of this is to show, you can see the spontaneous pulsation. So you can actually see the blood vessels pulsating in this lesion, and ultrasound is helpful for that, so that you can identify this as a melanoma, which is one of the few tumors that has its own blood supply that you can kind of see pulsating as shown in the video. Autofluorescence. These are some of the black and white pictures that we showed earlier. This is, you're able to detect lipofuscin, which we often call orange pigmentation. This is not only seen in melanoma, but it is a helpful differentiating feature. So here, you can see this two different lesions with different colors. So a pigmented melanoma up top and an amelanotic lesion at the bottom. And you can see at the pigmented one at the top, if you look very closely, there's some orange pigment over it. That orange pigment shows as brown on the amelanotic lesion, and the autofluorescence helps you identify the areas of lipofuscin as it lights up. And so that way, based on the color of the lesion, you're not necessarily confused as to whether or not it's lipofuscin, because the autofluorescence shows you that as it lights up. But there are other lesions where you can see the lipofuscin overlying it, and so as I said before, it is not 100% diagnostic for melanoma, but it is helpful. And you can also identify the fluid that is developed from these lesions. In the color picture, it's really hard to notice or appreciate how much fluid there is, but the autofluorescence shows that well. OCT, one of our last modalities. So this is really, really helpful to identify if there's subretinal fluid, and also for small lesions that are not large enough to differentiate on ultrasound. This gives you some different characteristics here. So number one, looking at the fluid. You can tell a little bit about the way the fluid looks and what the retina looks like around it. You can get an idea of how long the fluid has been there. So up at the top, you have a lesion that has some fluid around it, and it's kind of subtle or hard to see, but the retina on the outside, the photoreceptors or the cells there, they don't look so great, and that's because the fluid has been there for a really, really long time. And usually that suggests that, again, we talked about the signs of chronicity, that goes along with a more benign lesion. Versus on the bottom, you can see that there's some shagginess to those cells, and that indicates that the fluid is more recent, more likely to be a melanoma as the fluid just developed. There are other lesions that you can see on OCT and have different characteristics. So if you look just at a color picture of the lesion at the top, you might say, well, that might have some pigment to it, I'm not sure. When you look at the OCT, you can see that it has kind of a lumpy, bumpy appearance, which is more typical in metastatic lesions, in cancer that's originated somewhere else in the body and then traveled to the eye. And this lesion here is an area of bleeding, and you can actually see the blood pushing everything up as opposed to being kind of a solid mass. And so, again, each of these individual characteristics or individual imaging modalities don't give you the diagnosis, but they are helpful in differentiating what the lesion of origin is. And so, again, up top you can see metastasis with that kind of lumpy, bumpy appearance. Fluorescein angiography, or ICG, this is where you get a dye injected into your veins, and then we take pictures of the eye to see what's going on with the blood circulation. This is not something that is really helpful in terms of diagnosing melanoma, but it helps rule out other lesions. So it helps us identify that, that something is not a melanoma. So here, for instance, you have a hemangioma, and it has the characteristic appearance of, on the bottom, lighting up quite early, and then over time, that washes out as the blood vessels of the choroid wash out as well. So what about biopsy? You can do biopsy for cytology, and that allows you to diagnose whether or not a melanoma is present or whether a carcinoma is present. And these images were partially courtesy of Dr. Harbour from when I was a resident, and from Dr. Augsburger when I was in Cincinnati. And the first thing you want to do for these biopsies, you want to identify where the lesion is. And so doing transillumination, unfortunately the video doesn't work, but basically you can put a light on the eye, and that will give you the outline of where the tumor is, and then you can use a marker essentially to mark around the area of the tumor, and then you can approach your biopsy from there. More recently, through work that Dr. Harbour talked about earlier, we know that there are a lot of mutations seen in this tumor, and I'm not going to talk about the molecular prognosis in this, but I am going to talk about the fact that now using various modalities of testing, including Castle Biosciences, you can identify certain mutations in the lesion. So when you get a biopsy, this is currently not used right now to determine that a lesion is a melanoma, but it is helpful when you do a biopsy to see a G protein mutation and to see a secondary mutation, because that helps confirm or reinforce that it is a melanoma. If you don't see it, maybe you just didn't get enough tissue, or maybe it wasn't shown in that biopsy, but if you do see it, it can be helpful. So we'll see if the video plays here. Okay, so this is kind of what a biopsy looks like in the operating room, and you can see us withdrawing. The tubing is connected to the needle here, and so you can see, and we'll do a close-up, so the next video will be a close-up as a heads up, right? So you can see what it's actually like to go into the area of the tumor. And then as we're on our way coming out, we use cryo or a freezing treatment so that no cells are coming out at the same time we do this procedure. Here's a close-up view, and we kind of have the eye fixed in position. Oh, you got spared. So I didn't show the biopsy here, but essentially the needle goes in at that angle for tumors that are more towards the front part of the eye. When the tumor is further in the back part of the eye, though, you can't go directly in because you can't rotate the eye that much. So instead, you have a needle going more towards the front part of the eye, and then it angles to the back. Sometimes we do this with the headlight over, or sometimes we use the microscope. And here, the video won't play, but here you can see with the microscope, you can see the tumor here, and then you can identify an area that doesn't have much in the way of blood vessels, so you can bring the needle in that direction. There are downsides to doing biopsy or potential complications, and patients ask about this all the time. One, and the most common, would be bleeding. So here's a patient that underwent a biopsy, and you can see that after the biopsy, the view was a little bit more hazy because there was some bleeding in the eye. But the vast majority of time, the blood clears on its own. So it tends not to be something that is particularly concerning, although a small group of patients do require a surgery to clear out the blood. The vast majority, it just goes away on its own. And everyone asks about the risk of tumor spreading outside the eye. There are very few reports of this, but there are some. With modern techniques and through some of the advancements of Dr. Jerry Shields and Jim Augsburger, as Dr. Harbour mentioned earlier, we do this very, very safely. And so thankfully now, this is not a major concern for spread outside the eye. And so we're able to get good information for diagnosis and for prognosis, as Dr. Correa will talk about in the future. So highlighting just the three areas: iris melanoma, the front part of the uvea. We can often see these when you're looking in the mirror. You can see that kind of in the front part of your eye. There's a number of characteristics that we look for, but iris melanomas in general have the best prognosis. Perhaps that's because it's in the front part of your eye. You can see it fairly early and they get treated. Perhaps they have a completely different makeup from other tumors. There is some information that suggests that even tumors that show as aggressive in the iris, they don't behave as aggressively as they would if they were in the choroid or the ciliary body. And so here's a number of different types of tumors of the iris that you can have. And here's actually a patient who had a nevus on the iris for a long time and developed that little knob in the middle of the melanoma. They were treated with a plaque, and you can see that it went away quite nicely, and you can see that specifically for the melanoma it went away, and you can kind of see before what the lesion looked like many years before, and so you can see that the nevus component of it, or at least the flat component of it, didn't really change with radiation. Ciliary body. So these lesions tend to have a worse prognosis, and you can kind of imagine why. You can see on this pathology slide after the eye has been removed that it is grown in an area where if you're looking through the pupil, it's really hard to see. It's not in the front of the eye, and it's hard to see in the back. By the time it's grown enough for you to identify it, it tends to be fairly large, and in this image you can see it's even pushing on the lens a bit, which can happen. And so there are some characteristics that you can identify. Again, any patient that's looking in the mirror, you would be able to see this area and know that something is not right. However, it can be more subtle. You can see areas of prominent blood vessels that are there, and that can be because there's a tumor underneath and those blood vessels are feeding it. And so this is something that we think about when we're examining. Now, there's a number of treatments for uveal melanoma. The vast majority are treated with radiation, most commonly plaque brachytherapy here in the US, and a small percentage are treated with enucleation. Primary cure rate, 95 to 98% in the vast majority of these cases, and we'll just quickly walk through the steps of these procedures. So again, you can see transillumination, the light on the eye to identify the area of the tumor, and then you place some marks where the tumor is. Then you take a dummy plaque, basically just a clear area, so you can use that to identify where you want to place the marks for where the plaque is going to go, and then you place your active plaque in place. And then you can do an ultrasound to confirm the positioning of the tumor. And I think one of the first papers on this was by Dr. Harbour back in 1996, and this helps us to know that the radiation is exactly where we want it to be. If you do that at the time of placement of the plaque and at the time of removal, you can also see that there's been no shift over the course of the few days where the plaque has been in place, and you can see that there's no swelling or bleeding that caused displacement of the plaque either. And you can see very, very good responses of the tumor. Proton beam radiotherapy, usually a surgery is involved where they place tantalum clips, again, to identify the location where the radiation is going to be performed. And then you're kind of fixed in the machine, and then the radiation goes towards the eye that way. And you can see kind of how small the tantalum clips are there next to the ruler. And lastly, I will go over some of the steps of enucleation. A number of patients have asked me about this before. Again, you can close your eyes if you don't want to see it. They're just photos as we go through. And so you can kind of mark the area where the conjunctiva is as you separate it. And I actually looked here, I don't know, Dr. Correa may have performed this surgery. These were from Dr. Augsburger's pictures. And so you can isolate the muscles, and so once you have them all attached to the sutures, you can separate all the muscles, and then you cut off the last two, and then you remove the globe in one. And you try and get as much of the nerve as you can, because in the event that there's any disease near the nerve, you don't want to leave any tumor present. And that's the orbit behind the eye, and that's where you're going to put the implant. There's a number of different types of implants that you can use, and on the top left you see some sizers, basically to make sure that you have an appropriate size of the implant that you're going to put in place. In the upper middle, you can see that there's a peg where basically we used to think that if there's a peg there, you get better movement, but we know in situations of cancer, you don't want to have a metal object in the eye because that prevents your ability to do MRIs in the future. So it's not really something that's used anymore. And then we put the implant inside, and then we attach the muscles. And there's a number of ways you can do this. You can attach the muscles to each other, you can attach the muscles to the implant, or if you cover the implant with scleral tissue, you can attach the muscles to that tissue. And once it's sutured in place, you close everything up, and then you put a little clear plastic shell there in front. You allow the eye to heal, and then once it's healed, you can go to an ocularist and have a fantastic prosthesis put in. And as you can see here, it can actually be really, really difficult to tell which eye is enucleated. And the motility or the movement of the eye is also pretty good. So there's a number of different types of implants, and it depends on where you are and what the surgeon preference is as to what you'll get. But the picture on the top left, you can see, has some grooves, so you can actually stitch or attach the muscles to that. The picture on the bottom left, you can see the implant going into donor sclera, and so that way it's wrapped kind of similarly to how your eye is now, and you can stitch to the donor sclera. And then I'll just briefly talk about surveillance, not so much in terms of following patients long-term, but knowing what we do up front. So we have often patted ourselves on the back as ocular oncologists saying that there's really good cure rates locally, but we also know that metastasis is obviously something that we're paying most attention to because that's the real battle. Even though there's a high percentage of patients, or potentially high percentage, that can develop metastasis, only a small percent develop it in the beginning. And Dr. Harbour mentioned about micrometastasis before, and so we know that some of those micrometastases are not going to be detectable on imaging up front. However, we do want to identify this in the group of patients at the diagnosis that do have it. So we'll get imaging at that time. Everything after this point, after your diagnosis, after your baseline testing, after your treatment, falls under the category of Dr. Correa's talk in prognostication, and then what happens after radiation. So that's just the basic Ocular Melanoma 101, what we're thinking about when we're seeing patients up front, the reason why we get so many pictures, the reason why you hang out in the office for so long. So thanks so much for your attention. I greatly appreciate it.
Thank you so much, Basil. And again, we will hold questions till the end, and then we'll break for lunch. So our next speaker is Dr. Zelia Correa. Dr. Correa is a professor of ophthalmology and co-director of the Ocular Oncology Service at the Bascom Palmer Eye Institute in Miami. Dr. Correa has dedicated her career to improving outcomes for patients with eye tumors by championing translational research and evidence-based care. She is an ocular oncologist and has made significant strides in the diagnosis and prognosis of ocular melanoma tumors. And again, you can read her full bio in our program, so please join me in welcoming her.
This is a true pleasure. Thank you so much for the invitation, Dr. Selig, and it's great to see a lot of colleagues here, but also patients, patients that I care about every day and that I love so much. My career spans for over 25 years, and it's been really a joy to share the journey of so many people and be part of their life. So my role here is to talk about prognostication and ocular care after the treatment of uveal melanoma. I am the Director of Ocular Oncology and Echography at Bascom Palmer Eye Institute, and I do consult for Castle Biosciences and the gene expression profile test. So what is the importance of prognosis? It's a big word, prognosis. What it means really is that you are trying to estimate which patients are at risk for having problems after we treat the primary tumor. As Dr. Williams, Dr. Harbour have told you, the cure rate for the primary tumor, meaning the ocular tumor, is very high. However, over the years, we haven't made a lot of progress in terms of, number one, identifying the people that were at risk, and you will see why. And number two, we didn't make a lot of progress in treating the metastasis, probably because we didn't understand the disease, and we didn't understand that gap between what happens from the eye tumor to the liver. So that's what we're going to talk about. The importance of this prognosis is to allow us to prepare the patients for potentially bad outcomes, but also to ensure healthcare resources are better distributed and we focus on those that really need it. It also is important for us because it allows us to see who are the patients that will benefit the most from what we call the adjuvant treatments and clinical trials, and empowers the patient to make treatment and life decisions. So for many years, estimating prognosis was based on clinical factors. First, we thought, older patients will do worse. There's some people that thought men would do worse than women, and we very quickly found out that that was not going to work. Then we looked at larger tumor diameter, thicker tumor, ciliary body location, diffuse tumor, and all other characteristics. And also, we weren't very accurate. It was better than the other clinical features, but it wasn't really that accurate. Then we started looking at tumor cells, and we were trying to look at, how the tumor looks under the microscope and what are the features around the tumor and all that. But that very quickly was no longer useful. As we move from removing the eye, we move into preserving the eye and trying to preserve vision. So then we looked at cytogenetic features, and it all started with identifying the loss of chromosome 3 or monosomy 3 was a risk factor, then to chromosome 8q gain or 8p loss, and then chromosome 1, chromosome 6. But even when we plotted that, it just became very complicated and not very accurate. And then finally, we got to transcriptomic features and gene expression profile, which was the work that was developed by Dr. Harbour, and I was very proud to be part of the validation study, the COOD-1, way back when nobody was doing biopsies on all the patients. And we really feel that that was a game changer. So if we look at CancerNet, you will see that the 5-year relative survival rate for small choroidal melanomas is 84%, but that very quickly declines when you're looking at larger tumors. And this is also a manner of prognostication. Most of you have heard of the American Joint Committee on Cancer, AJCC, and they come up with this classification every few years. They review it, and when you look at it, it looks pretty convoluted. It reminds me of that very fancy new museum they have up in New York, I think, or DC, that has like a bunch of different staircases and all that. But if you're in the office and you're in front of your patient and you show that, they're like, okay, we're playing puzzle now? Is this what we're here for? And it is a good classification. However, the problem with that is that they grabbed over 1,000 patients that they already knew the outcome of, and came backwards and tried to create a classification that would fit all of them. That's why it looks like a staircase. So that for clinic and for patients is very complicated. I'm not a big fan, although I have to put that in because the AJCC said that we do need to do it. And so this is how it looks with the stages and the patient survival over there. So you can see that stage 1 patients have a much longer survival or a lower development of metastasis compared to patients that are stage 3 or larger tumors and so forth. So for a long time, you know, when I was in Cincinnati working with Dr. James Augsburger, who was my mentor, we really wanted to look at these things. We really felt like being able to identify the patients that were at high risk for development of metastasis would be a game changer. And it would allow us to have early intervention and hopefully really change the outcome of these patients. So a long time ago, well, 2017 doesn't seem like that long, right? We looked at the comparison of alternative tumor size classifications, which was what we used and what the collaborative ocular melanoma used and a bunch of the other studies, to look at how well they were able to predict development of metastasis. And then, you know, around that same time, Dr. Harbour published on GEP and PRAME status versus tumor node biopsy or tumor node metastasis staging for prognostication of uveal melanoma. And so what we saw was that really all of those very, very elaborate classifications that you're seeing here, the separation of the curves was best when we were looking at simple linear diameter of the tumor or the cubic volume, or then that very fancy stairway kind of thing. And the prognostic significance, now this is the shocker, is equally good if you were using what we call the TNM classification, which is the stairway, versus looking at the linear basal diameter alone. So this is what we did. I mean, this is a mathematical exercise. Don't want you guys to think about it or try to understand it. But essentially what we wanted the 3 curves to be as separate as possible, because that would really indicate, okay, I am identifying and I'm placing the patients in the right location here or the right prediction. And you see here that the TNM classification, when we had the 4 classes, was discriminating, but it was better when we kind of got together the type 2 and 3. But when you look at linear diameter alone, was almost the same. You can almost compare and overlap these 2 curves. So that was quite shocking. It was an interesting finding for sure. And what we found out is that in the office or for a patient, if you're really interested in thinking about this and thinking about your outcome clinically, it seems like the simplest classification or the linear largest basal diameter, we call it LBD. We have so many acronyms, but linear largest basal diameter is just as good as the most complex classification that you can see. So what about gene expression-based classification? As I mentioned to you back in the 2008, 2009, when we started using gene expression profile as a research tool to try to correlate with development of metastasis, we didn't think, or maybe Dr. Harbour did, we didn't think it was going to get to be this big and this helpful to us. But anyway, what we know today is the class 1 PRAME-negative patients are the lowest risk, and they have this driver mutation that we call EIF1AX. And this mutation is a mutation that basically guarantees the patient won't have the other 2 mutations that are progressively worse. So class 2 PRAME-positive patients, which are the middle of the road risk, they have this splicing mutation. It's called SF3B1. And if you guys want to take a picture of that, it's quite interesting just to have that knowledge and that information. And then the patients that are at higher risk are the ones that have class 2 PRAME-negative or class 2 PRAME-positive. And these patients, they do have a BAP1 mutation, which seems to us nowadays to be really the culprit and the game changer in terms of risk. So when we look at 240 patients with uveal melanoma using the TNM classification versus the GEP PRAME, you see that, again, the curves almost overlap. But with the benefit that, you know, we see that GEP PRAME is much more accurate and it really matches better what we're predicting in terms of outcomes. Also, GEP shows us that, you know, patients that are at the lower risk, they really do very well in the first 5 to 6 years, but we know that this is a dip in the curve that all of us ocular oncologists have debated with, because there is obviously a significantly worse outcome in the first 5 years, but then the patients that do well thus far around year 7, there seems to be a slightly lower, you know, a slight dip, and we are trying to find out who these folks are. So when you see here the TNM classification with the 3 categories or the GEP PRAME with 3 categories, again, the separation between the curves is much better right there. So the conclusion here was that GEP PRAME both demonstrated a better prognostic accuracy than the TNM staging classification, and these findings continue to support what we've been doing. So another one that we also use is using the GEP, the gene expression profile, plus the diameter of the tumor. And this kind of may be getting boring, but this is to us in everyday practice very, very important. And what we found was that the patients that were GEP1 but were wide, were big tumors, they had a similar middle of the road outcome as the patients that had the GEP2 but were small tumors. So it seems like, although the size isn't the number one thing, but it seems like the size does help us a little bit in terms of evaluating these patients. And this again compared to the TNM classification once again. This is what we saw when we did the Collaborative Ocular Oncology Group 1. And at that time, we had nothing. We really had nothing. We were looking at tumors. We were trying to use the COMS classification. I remember that. It was a crazy, crazy time. Patients would look at me and say, what should I do? And I really didn't have a good answer. But now when we look back with the first attempts looking at chromosome status, we see that yeah, it does help a little bit, but it doesn't really make the same predictive negative predictive value, what we call it. So meaning the real separation between those that will do well and those who won't. And here you see exactly what I mean when we overlap the 2 curves. You see here GEP class 1 versus loss of chromosome 3, and here's GEP2 and loss of chromosome 3. So these are the early COOP results that we are looking to publish. I think Dr. Harbour already briefly mentioned that. So we're seeing that the class 2 BAP mutant and PRAME positive, they are at higher risk compared to the PRAME negative and compared to the class 1 tumors. And what we're seeing is a progression-free survival at 36 months of the class 2 tumors of 93% and of the class 2 tumors at 44%. And a lot of people also talk about the TCGA. The TCGA was a group, they collaborated, they got all of these enucleated eyes, and they tried to create a one-time classification or one-time evaluation, and incredibly, or surprising, or not surprisingly, they found things that were similar to what we all knew, including the EIF1AX mutation, the SF3B1 mutation, and the BAP1 mutation, as you can see right here. And the GP PRAME versus the TCGA, you see here that the TCGA was not really a classification, but it was a one-time grouping of 80 selected large tumors, and they do coincide with what we use as far as class 1, class 2, and obviously here would be the PRAME positive patients right there. So now that we've kind of gotten a little tired of this, talk about a lot of, graphs and a lot of strategies to how to look at this, I want to talk a little bit with you about ocular care after treatment, because to me this is equally as important. Why is it? Well, number one, because the ocular care is really your quality of life. It's your eye and how you're going to feel about it, how you're going to go about your life, because there is life after this kind of diagnosis, and we know that very well, right? There's a good life after that too. So ocular care is impacted by a number of things. Number one, what is the tumor location and the tumor size for those that do the plaque brachytherapy? How can we go about measures to avoid vision loss? How do we monitor tumor regression? And how do we monitor radiation side effects? And for those that have to have their eye removed, how do we monitor for the endophthalmic orbit or socket? And on the side of that, there is the surveillance testing, and I'm not going to go too much into that because I know Dr. Lutsky and the other doctors are here to talk to you about that, and they are much more experienced, and they will give you the other side of it. And it's also determined by the transcriptomic profile, the surveillance testing, and the high-risk patients are followed by the oncologist. So that's another thing. I personally don't try to do surveillance testing for high-risk patients. When the patient has a low-risk profile, we may order the tests in the office just to make it easier, and obviously make sure the patient has access to this kind of surveillance and not overwhelm the oncologist. But anytime the patient has a high-risk profile, it's important to send it over to the oncologist. So, impact of tumor size and location is really looking at posterior versus anterior, what we call the tumors in the way back in the eye, is close to the optic nerve, is close to the fovea, or is it on the front part of the eye, as you can see here, away from the nerve, away from the focusing point. And the tumor size, because larger tumors require more radiation. So even if the tumor is not very close to the back of the eye, if it's large, it will get almost as much radiation to the focusing point in the nerve. And that's why when you're in the office and you see the doctor measuring this and measuring that, we're all kind of already factoring in all of these variables and thinking about what we need to do next and how can we keep you the best way we can afterwards. So the measures to avoid vision loss essentially now are surrounding anti-VEGF for tumors that are located within what we call 3 millimeters. And 3 millimeters is about 2 little color pinheads that we use for sewing and hemming. Put them side by side, that's 3 millimeters. That's how close it is. So Dr. Ivana Kim championed the use of anti-VEGF in a prospective way, randomized way, which is the right way for us to do our research. And she actually measured by what we call disc diameters, but we know the optic nerve or the optic disc is about 1.5 millimeters in diameter. So it's 3 millimeters. And the question is when to start, how often, and how long. So there are some doctors that do start the anti-VEGF, the vascular endothelial growth factor, at the time of the plaque therapy. I don't, because I feel that all of that radiation, inflammation, and all that chaos that happens in the eye from the radiation is important to kill the tumor. So it's like, well, I need to let the tumor be exposed to all that inflammation to have a good tumor regression. So I personally start at 2 months, and how often? Every 2 months, as Dr. Kim showed us, and for how long? For 2 years. But there are variances in that protocol, and obviously your doctor will have their rationale. Important to ask them about it. So, important to always monitor visual acuity and ocular pressure. Although sometimes, and I tell the patients, be very aware that even if you lose some of your central vision, the peripheral vision means a lot. Because the peripheral vision may be your ability to drive. It may be your ability to look at the side mirror here when somebody is coming around your car, or your ability to go up and down steps. So don't underestimate the peripheral vision and how much it impacts the quality of life. Another thing that is very important is ocular pressure. Sometimes we're so focused on the tumor, we forget that ocular pressure alone may damage the nerve and decrease the vision. So ocular pressure is very, very important. And the ultrasound to measure the tumor. And sometimes I don't measure the tumor every time the patient comes in, especially after we've gone through that first year. So I look at everything is good. I may not repeat the ultrasound every time. And sometimes the patient says, well, aren't you worried about my tumor? And I say, well, but I can see it, and I see that it hasn't changed. I don't have to put you through that every single time. But we do like to use this OCT, that optical coherence tomography, and I think that those of you that have seen it on the screen where your doctor says, oh, there's more fluid, there's less fluid, it's getting better, it's getting worse. So this OCT really is a very interesting technology and allows us to see even very tiny changes before the patient manifests a change in the vision. So that's very, very helpful. And evaluate development of cataract, because this is probably the #1 side effect of the tumors in general, is that most patients that have the radiation will probably develop a cataract within 2 to 3 years by, on average. And the additional treatments that may be needed. And that includes adjuvant laser around the tumor margins if there's still fluid, or if the doctor feels that maybe that area is a little suspicious or not well treated, periocular and intraocular injections for inflammation, swelling of ocular tissues, and cataract surgery, other lasers, other surgeries if there's a retinal detachment or if there's bleeding in the eye. All of those things can be done. So monitoring the endophthalmic socket is very important because, #1, the cosmesis, nobody wants to look like they have an artificial eye. Follow up with the ocularist is very important for cleaning, for maintenance, for adjustment. The socket, the lids, as we age, it changes, and obviously the same way, the kind of fitting will change. So make sure that you do that. Monitor the lid appearance and discharge. Most patients that do have an artificial eye will have more discharge. And that's probably because we're going to have a greater surface of conjunctiva, that lining, which is the lining that really kind of modulates the allergic responses. When you get a cold, you get more discharge, etc. So monitoring that is very important. And observe the conjunctival surface underneath the prosthesis to make sure that it's nice and smooth, it's pink, it's not red, and there's no exposure of the implant. And the surveillance testing, as I said, directed by transcriptomic and genomic testing. I'm going to let Dr. Lutsky and his colleagues talk about that. High-risk patients should be monitored by the clinical oncologist and melanoma specialist. And then mental and emotional health. It's very, very important. We don't talk about it enough, right, Sara? We just don't, right? It's kind of like we're so focused on the science. But, this is something that at Bascom Palmer we're looking to start a program to give support to our patients. There's very limited discussion about the topic. There's no protocols on how to approach the patients. And the benefit of therapy and support groups has not been established, but it's incredible how much patients feel better when they have other people that are in the same place as they are that can share their journey. And taking care of your health. I have a lovely patient here, Kyleigh Ashton, that was the first one that said to me, you know what, I'm going to go all out there. I'm going to change my diet. I'm going to I'm gonna detox and everything, and how wonderful has it been, right? So interestingly enough, healthy eating habits really improve your overall immune system. It also allows your immune system to better respond to any immunotherapy that can be attempted in case you do develop metastasis. Other things, eating healthy foods obviously is significant in a sense of your gut microbiome. So a recent study has shown that American patients, they have a much worse gut microbiome than European patients. And because of that, they dare to say that maybe our patients are developing more metastases than the European patients because they're not eating as much processed foods and all the toxic stuff that tastes so good but does so bad for you. You know, obviously everything that tastes good is bad for you, right? So, and other things, exercising, sleeping well, and all of it really plays a significant role. So the take-home messages here are, number one, prognosis of uveal melanoma is still a topic of discussion for some physicians. Clinical factors have shown to be inaccurate prognosticators alone. The AJCC or TNM classification is pretty cumbersome and complex, and most ophthalmologists won't be using that in the office. It's really, really hard. GEP associated with PRAME status has shown to be the most robust and predictable. Monosomy 3 has also shown to be fine, but it's less predictive than GEP because of some science stuff that we don't need to get into. And GEP with the largest basal diameter has also shown to be prognostically superior than TNM. After that, I want you to remember that follow-up after treatment is important and consists of ocular and systemic monitoring. Discussing the options of radiation side effects, of treatment of radiation side effects with your ocular oncologist is very, very important for your quality of life. Remember, you know, after the treatment, there's a whole life after that. And everybody talks about the first 5 years, and sometimes you have to think about the next 10, 15, 20 years. And that's a difficult concept to grasp. Ensure you ask questions about your vision prognosis and about new treatments or clinical trials every time you go to your doctor. And take care of your systemic and emotional health. And that's all I wanted to share with you today. Thanks again.
Thank you so much, Dr. Correa. You can actually have a seat right up here, and I will just request that our other two speakers come back up. And we are going to open up the floor to some questions. You guys can just sit right here. And I'm going to give these to my colleagues, and they will run around. And we will have about 15 minutes for questions before we break for our lunch. We're going to take the photo first, and we're going to hear a little bit about our Miles for Melanoma events and a local event here hosted by some of our advocates. So we can start off with questions right here.
Hi, good morning. Thank you for your presentation. I'm the new kid. I was diagnosed in October of last year. My cancer testing came back PRAME, class 2 PRAME positive. And I do not have the HLA protein. But my question for you too is I have psoriatic arthritis and psoriasis, and I've been on a biologic since 2016. I've been on several. Most recently Humira for the last 2 years. I can't get a straight answer on the biologics and how that could play into the disease. So I just wanted to see if any of you had anything to add to that.
You know, this is a tough question, but I think this may be a better question for the medical oncologists. You're going to have a panel like this in the afternoon, right? Ma'am, could you again in the afternoon? We're the eye folks, and we have some world-class medical oncology folks here who will take that question.
I have a question, maybe while others are thinking about their questions, but I know a question that we get a lot from patients is that if they didn't have an opportunity to have a biopsy before the eye was treated, so much of what you've talked about relies on, you know, biopsy outcomes. So how would you advise that patient?
if they haven't had the biopsy, I tried to get their records and I used the largest basal diameter. Largest basal diameter. I think that would be the only clinical factor that through the years we have shown that it has some, correlation with outcome.
And any, well, I guess Dr. Harbour, you spoke to this a little bit, but thoughts about the possibility of obtaining a biopsy post radiation or other ways to get some of that genetic or molecular information?
Yeah, I mean, there are a couple of things. If the radiation has not been too long ago, within a few months, you could still, the RNA is likely to change, but you could at least get a biopsy for mutations and look for chromosomal changes, because those are DNA changes. So you could do that. The other thing that I think we're not very far away from is what I touched on with circulating tumor DNA. And I would first agree with Dr. Correa that we can use the clinical criteria, the diameter, and we can use the TNM classification to approximate as best we can the risk level of the patient. And then I think in the near future, we can't use circulating tumor DNA to replace tumor biopsy, but at least we can monitor, in the future, in the near future, we'll be able to at least monitor for early signs of, tumor progression.
I was just going to add that I agree with everything that's been said, but I think, one of the benefits of being a patient at this meeting is you have access to other patients who have been through it, and you have access to a lot of information. It is a little bit more challenging if you're seeing your ocular oncologist and you're not offered a biopsy in the beginning and you don't know about a biopsy, because then, oftentimes you miss the window for the biopsy up front for molecular prognostication. And we recently worked on a paper with Kyleigh and Sara kind of looking at this, and it seems to be a small percentage of patients that don't get offered biopsy, but this is an opportunity for patients to advocate for themselves and to bring it up if it's not something that's discussed. And that's why this meeting is so important, so this kind of information can be disseminated.
Yeah, we have a question in the back and then up front here.
Thank you. First of all, great talk, so thank you so much to all for those. My name is Matt Murphy. I'm actually a palliative care doctor down here at Moffitt. So nice to see some of my Moffitt colleagues here, and look forward to connecting with everybody. And I'm also the son to a father who was recently diagnosed with uveal melanoma, and actually was immediately referred for enucleation with Dr. Carol Shields. And based on the karyotype of the specimen, he was deemed to be in the high-risk category based on monosomy 3 and amplification of 8q. And we've heard so much this morning about some of the interesting work that some of you have done in terms of further risk stratification and prognostication. And so I guess my question is specifically, I wasn't told anything with regard to PRAME status or any additional testing, really just the karyotype information. So I'm wondering at this point if any of you, is there utility to other testing on that surgical specimen, or is what we have enough to kind of have the most helpful information? And look forward to maybe connecting with some of you offline as well outside the Q&A. Thank you.
Yeah, so, most of the time, if you're monosomy 3, you're going to be class 2. And, if you have, the thing with 8q, it's more quantitative. If you have 1 extra copy of 8q, it doesn't really help you. But if you have 3 or 5, extra copies, then that's more of a poor prognostic sign. So it's just a little squishier with the chromosomal data. But you can certainly, it's certainly more useful than just going by TNM data. So I think that, that what your father has had so far is certainly very helpful. The capsule test can actually be done on paraffin-embedded tissue. So if there was desire to have further information, you can request that it be done from the surgical specimen, which, you know, with a needle biopsy, you don't have that, but if there was a nucleation, you can ask for that. And the PRAME is part of that test, you know, and actually the mutation panel is also part of the test from the same section. They can get all of that information. And, you know, most of the time they're going to be in agreement, but where there's disagreement, usually the gene expression based data is going to be more accurate. So, you know, I would probably want to have that information if possible.
Hello, I'm interested in the database of biopsies you all have collected to date, how many biopsies you have, and what the next step in your research will be going forward with these class 2 gram-positive tumors that you've biopsied. I know you've talked a little bit, Bill, about an adjunct trial. Could you tell us a little bit about what's the next most exciting adjunct trial you see coming down the pipeline?
Sure, so the COOP2 study, the Collaborative Ocular Oncology Group 2, which we were all 3 a part of, and 22 other centers, collected samples on over 1,700 patients prospectively and performed this entire genetic profiling that you heard about. That's never been done before in melanoma anywhere. So we are mining that data set now. The first paper is being written now. That we showed you a sneak preview of some of that data, but it's going to go into a lot more depth. And there will be a series of papers that come forth from that. It's a lot of data to mine that we'll be mining, and you'll start to see the papers coming out very soon for the whole data set. And then the beauty of this, it'll become really available to the whole group to mine in different ways, you know, to look at different subgroups. You can ask a number of different questions about, you know, that will really, this data set will be used for years to come. To me, the next step in this progression, which, you know, this line of work is really twofold. One is using tumor biopsy to determine which small tumors to treat. You've heard now quite a bit this morning about small tumors, the desire to, you know, if you really do have a true melanoma, to try to treat it when it's small before it grows, but having some difficulty just looking at clinical criteria to determine that. There's increasing information. We have about 500 patients so far where we've done the needle biopsy in suspicious nevi. And it's extremely helpful in that situation in distinguishing melanomas from nevi. But as with all the work that we've done, I think it's important to have prospective multi-center validation. You know, a lot of the things that are out there, the TCGA classification, the chromosomal testing, none of those have been prospectively validated. So, you know, this is you know, we have a separate data monitoring group. It's prospective, it's multi-center, so you can really feel good about the quality of the data when it comes out. So while we're increasingly using the biopsy in that setting, we really want to prospectively validate it. The other branch, I would say, is liquid biopsy. You know, what's going to be the role, both ocular liquid biopsy, as I mentioned, and you know, blood liquid biopsy. What's going to be the role of those in prior to treatment of the primary tumor and after the treatment of the primary tumor? So I think that's where this line of research is going.
Yeah, I think of all Dr. Harbour said, I just want to reiterate to everybody that while liquid biopsy is going to be very helpful and it can be the way of the future, but you know, getting aqueous and vitreous is not going to be as accurate as getting the tumor tissue. So that's important for patients to understand, that these can be surrogates, but they're not going to be because we're not really going to be grasping the tumor cells where we can study in greater depth.
All right, we are at time for questions. However, our wonderful panel is going to join us for lunch, so if you have any other questions, please feel free to ask them, and please join me in thanking them for 3 extraordinary pieces of research.